Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. My name is Rizka Anissa Zahira. It's a precious chance for me to be your master of ceremony on this very special occasion, the second Sriwijaya International Conference on Basic and Applied Sciences 2021. Ladies and gentlemen, allow me to take this opportunity to welcome all of participants of this conference by first offering praise and gratitude to the presence of God Almighty for all His blessings and His graces so that we can gather here in this online room in excellent condition and health. His Excellency, the Rector of Sriwijaya University, Profesor Dr. Insinyur Haji Ani Sagaf, MSCI IPU, first, second, third, and fourth Vice Rector of Sriwijaya University, Dean, and the Director of Postgraduate Program of Sriwijaya University. Respectable our keynote speakers, Professor Walid Fuad from the American University, Cairo, Professor Puji Lukitawati Haryani from Sriwijaya University, Indonesia, Professor Marcelo Ornagi Orlando from Sao Paulo University, Brazil, Professor Fazilah Adiba Abdul Majid from Malaysia Terengganu University, Malaysia, and Professor Arif Gusnanto from University of Leeds, United Kingdom. Respectable our invited speakers, Professor Zailan bin Siri from Malaya University, Malaysia, Dr. Dede Heri Yulianto from the National Research and Innovation Agency, Dr. Yosi Andriani from Malaysia Terengganu University, Malaysia, Dr. Endang Linirin Widyastuti from Lampung University, Indonesia, and Mr. Acep Furkon, PhD from Bandung Institute of Technology, Indonesia. The Honorable Dean of Mathematics and Natural Sciences Department, Dr. Hermansyah. Welcome to all participants of the conference present today. Ladies and gentlemen, on this occasion, allow me to introduce you today's agenda. Opening, a traditional dance performance from South Sumatra, Indonesian National Anthem, welcoming speech, opening speech, plenary session, parallel session, and the last is closing. Ladies and gentlemen, first, let's watch the traditional dance of Gending Sriwijaya from South Sumatra.
Dahulu kala berdiri teguh, kokoh sepanjang masa. Memasyurkan Indonesia di tanah Asia. Melambangkan keagungan sejarah Nusa dan bangsa.
Ladies and gentlemen, the next agenda is to sing Indonesia Raya Anthem. To all audiences, please stand and sing. Audiences are allowed to sit back. Ladies and gentlemen, now we invite Dr. Hermansyah as the Dean of Mathematics and Natural Sciences Department, Sriwijaya University, to deliver his welcome speech. To Dr. Hermansyah, time and place are yours.
Bismillahirrahmanirrahim. In the name of Allah, the most beneficent, the most merciful. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Rector Universitas Sriwijaya, Profesor Dr. Insinyur Haji Anis Sagaf, MSCI, IPU, the Honorable Vice Rector First, Second, Third, and Fourth, Universitas Sriwijaya, the Honorable keynote speaker and invited speakers, the Honorable Deans in Universitas Sriwijaya, distinguished delegates, ladies and gentlemen, good morning. It's my pleasure to welcome you all to the opening of the second Sriwijaya International Conference on Basic and Applied Sciences, which we held virtually through Zoom meeting. This is the second series of conference after in 2018, the first Sriwijaya International Conference on the Basic and Applied Sciences was successfully held from 6 to 7 November 2018 in Palembang, Indonesia. Despite the difficult situation associated with the spread of COVID-19, the interest in the conference is quite considerable. I am delighted to see that the conference speakers, either keynote invited or participant, present their latest scientific results, exchange experience, and discuss issues of mathematics and science, including the application field. And the conference adopts a time theme, science development in the new era, triggered by coronavirus pandemics. More than 300 participants attend to this conference virtually through Zoom meeting and YouTube channel. They will hear informative information from the 112 presenters from 6 countries, from Brazil, Egypt, United Kingdom, Japan, Malaysia, and Indonesia. On behalf of the Faculty of Mathematics and Natural Science, I would like to take this opportunity to express our sincere thanks to Rektor Universitas Sriwijaya, Profesor Dr. Insinyur Haji Anis Sagaf, MSCI, IPU. We would like to request Rektor Universitas Sriwijaya to officially open this conference. I would like to take this opportunity to speakers about keynote and invited speaker. The keynote speakers are Professor Marcelo Ornaghi Orlandi from Department of Physics, Sao Paulo State University, Professor Dr. Fadila Adiba Abdul Majid from Institute of Marine Biotechnology, University, Malaysia, Terengganu, Professor Dr. Puji Lukitawati Haryani from Department of Chemistry, Sriwijaya University, Associate Professor Walid Fuad, PhD, from Department of Biology, the American University in Cairo, Associate Professor Dr. Arif Gusnanto, from School of Mathematics, University of Leeds, UK. And the invited speakers are Dr. Dede Yulianto from LIPI, Dr. Acep Purkon from ITB, Dr. Sarifah Nuriza from University Malaya, and Dr. Yosi Andriani, HS from Institute of Marine Biotechnology, University Malaysia Terengganu. And I would like to thanks to the Dr. Mr. Muhammad Said and all members as the conference organizing committee for their diligent work and their contribution for this event. Thanks to Institute Marine of Biotechnology, University Malaysia Terengganu as a co-organizer of this conference. And in the closing, I hope that all of you will enjoy the conference, and I wish you every success with this important conference. Thank you. Billahi taufiq wa hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Dr. Hermansyah, for your delightful speech. The next agenda would be an opening speech. Allow us to invite Professor Dr. Insinyur Haji Anisagaf, MSCE IPU, as the Rector of Sriwijaya University, to deliver his opening remark. Please welcome Professor Dr. Insinyur Haji Anisagaf, MSCE IPU.
Thank you very much, moderator. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah wassalatu wassalamu ala Rasulillah wa ala alihi wa sahbihi wa mawala. Wa la haula wa la quwwata illa billahil aliyyil azim. Allahumma shalli ala sayyidina Muhammad wa ala ali sayyidina Muhammad. Honorable Vice Rector 1, 2, 3, and 4. The keynote and invited speakers, Dean Faculty of Science and other deans in Universitas Sriwijaya, Director of Postgraduate School, Head of Research Center, Head of departments all over Sriwijaya University and other universities, and all the presenters of the papers for the conference. Especially, I would like to greet first Professor Marcelo Ornagi Orlan from Department of Physics, Sao Paulo State University, Professor Dr. Fazilah Adibah Abdul Majid, Institute of Marine Biotechnology, University Malaysia Terengganu, UMT, Professor Dr. Puji Lukowati Heriani, MSc from Department of Chemistry, Sriwijaya University, Associate Professor Walid Fuad, PhD, Department of Biology, the American University in Cairo, Associate Professor Dr. Arif Gusnanto, School of Mathematics, University of Leeds, UK, and invited speakers Dr. Dede Heryulianto from LIPI, Dr. Cecep Furkon from ITB and ITERA, Dr. Sharifah Nuriza from University Malaya, Dr. Yose Andriani from IMB, University Malaysia Terengganu, and Dr. Endang Lanirin Widyastuti from Faculty of MIPA, University of Lampung, and the committee led by Dr. Muhammad Said. And ladies and gentlemen, first of all, I would like to appreciate, to say good luck to the faculty MIPA that had this conference for the second time with the topic of Sriwijaya International Conference on Basic and Applied Science with the theme Skin Development in the New Area. This conference is very important for us as the member of universities. We have to discuss what we have done and what we will do in the future. We cannot do anything in the world by ourselves. We have to we have to share each other and discuss with other people from all over the world. This conference was mentioned the first time in 2018, but due to the pandemic era, the committee has delayed this conference until today. I'm very happy to see this conference was attended more than 300 participants. This is a very big amount of the conference member. Hopefully, this meeting not only just for the conference, but we can discuss each other. We have already have MOU with some universities all over the world, including University Malaya and University Malaysia Terengganu. I have signed the the MOU at the beginning of 2020. Hopefully it can work and join research, join conference, join publication, and share our knowledge, especially in order to answer the situation right now. COVID-19 has already changed our life, changed our tradition, changed our old fashion of education. We have to find the new system and the new advanced knowledge that can we share together in order to do our life better in the future. As a rector, I would like to have high thanks to all the committee and also to the speakers, to universities that has relationship with Universitas Sriwijaya. And we are open for you all to come here after pandemic COVID has finished to do the research together, whatever you want to do here. And also bring your students, including we will also send our students to your universities. Have a nice day, have a nice life by seeing Bismillahirrahmanirrahim, majreha wa mursaha inna rabbil ghafurur rahim. You have all a nice conference today. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Professor Dr. Insinyur Haji Anis Sagaf, MSCE IPU, for the remarks and for the opening of the conference. Give applause. The next agenda is documentary session to be guided by the IT team. To all audiences, please turn on your cameras and keep your smile. First slide. 1, 2, 3. Smile. Next. The second slide. 1, 2, 3. Smile. Next. One, two, three. Smile. Next. 1, 2, 3. Smile. Next. 1, 2, 3. Say cheese. And the last one. 1, 2, 3. Say cheese. Thank you. Ladies and gentlemen, before we started this event, let us pray in accordance with trust and respectively. May God make this rewarding event for all of us, and the event will run smoothly and conducive. To Dr. Adi Rahmat, time and place are yours.
Assalamualaikum warahmatullahi wabarakatuh. First of all, thanks to Allah subhanahu wa ta'ala who always given health, blessing and salvation for all of us so that we can gather here in this great event. Please allow me to lead this prayer session in Arabic and Indonesia. A'udzu billahi minasyaitonir rajim. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin, hamdan syakirin, hamdan naimin, hamdan yuwafi ni'amahu wa yukafi'u mazidah. Ya Rabbana lakal hamdu kama yanbaghi li jalali wajhikal karim wa 'azimi sultanik. Allahumma salli ala Muhammad wa ala ali Muhammad. Allahumma Rabbana taqabbal minna salatana wa siyamana wa ruku'ana wa sujudana wa qu'udana wa tadarru'ana wa takhassu'ana wa tammim taksirana ya Allah ya Rabbal alamin. Allahumma inna nas'aluka salamatan fid din wa 'afiyatan fil jasadi wa ziyadatan fil 'ilmi wa barakatan fi rizki wa taubatan qablal maut wa rahmatan 'indal maut wa maghfiratan ba'dal maut. Allahumma hawwin 'alaina fi sakaratil maut wan najata minan nari wal afwa 'indal hisab. Allahumma anfa'na bima 'allamtana wa 'allimna ma yanfa'una warzuqna 'ilman yanfa'una. Ya Allah yang Maha Kuasa, pagi ini kami berkumpul di gedung Pasca dalam rangka konferensi SICBAS yang kedua. Berkahilah acara ini dengan kebaikan yang tak berhingga. Ya Allah, Zat yang mutakabbir, anugerahi kami kemampuan berfikir, menggali ilmu, menyingkap tabir. Jauhkan kami dari sikap yang pandir. Ya Allah, Ya Aziz, Ya Ghafur, khilaf dan salah kami sungguh tak terukur. Di saat nasi sudah menjadi bubur, ampuni kami sebelum jalani alam kubur. Rabbana atina fid dunya hasanah wa fil akhirati hasanatan wa qina adzabannar. Bifadli subhana rabbika rabbil izzati amma yasifun. Wassalamun alal mursalin walhamdulillahi rabbil alamin. Assalamualaikum warahmatullahi wabarakatuh.
The next agenda is break time. To all audiences, please take a 5 minutes break.
Universitas Sumatera tempat ini yang merupakan terkenal dengan ikon jembatan Ampera dan sungai musik. Universitas Sriwijaya memiliki 2 kampus yang terpisah. Kampus pertama terletak di Kota Palembang yang diresmikan oleh Presiden Insinyur Soekarno pada tanggal 31 Oktober tahun 1960. Dan kampus kedua terletak di Kabupaten Ogan Ilir, Indralaya yang diresmikan oleh Presiden Soeharto pada tanggal 6 Maret tahun 1997.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera, rahmat dan keberkahan Allah SWT tercurah untuk kita semua. Universitas Sriwijaya adalah salah satu universitas negeri yang besar di Indonesia dengan student body 28.000 mahasiswa lebih yang tersebar di 10 fakultas dan program pasca sarjana, dengan visi menjadi perguruan tinggi terkemuka berbasis riset yang unggul di berbagai cabang ilmu, teknologi, dan seni. Saat ini Universitas Sriwijaya terus maju dengan peningkatan di berbagai bidang. Bidang akademik dengan peningkatan proses belajar mengajar yang berbasis IT, dengan masa studi tepat, cepat, dan qualified. Bidang riset membudayakan kegiatan riset yang unggul, melibatkan dosen dan mahasiswa di berbagai cabang ilmu, sains, teknologi, sosial, humaniora, dan seni yang didukung dengan laboratorium yang berstandar. Bidang pengabdian masyarakat membumikan hasil-hasil karya Universitas Sriwijaya untuk kepentingan umat, bangsa, dan negara Republik Indonesia, membantu pemerintah Republik Indonesia, provinsi, kabupaten, dan kota dalam segi perencanaan yang baik, pemberdayaan masyarakat, penggalian dan pengkajian sumber daya alam, dan penanggulangan bencana alam.
Mahasiswa Universitas Sriwijaya berasal dari berbagai penjuru tanah air dan mancanegara. Terdiri dari diploma, strata 1, pasca sarjana, profesi, spesialis, dan doktor. Fakultas Ekonomi, Fakultas Hukum, Fakultas Teknik, Fakultas Kedokteran, Fakultas Pertanian, Fakultas Keguruan dan Ilmu, Fakultas Ilmu Sosial dan Ilmu Politik, Fakultas Matematika dan Ilmu Pengetahuan Alam, Fakultas Ilmu Komputer, Fakultas Kesehatan Masyarakat, Program Pasca Sarjana X2, dan Program Doktor S3. Kerjasama internasional Universitas Sriwijaya diantaranya meliputi negara Thailand, Malaysia, Jepang, Vietnam, Jerman, Amerika, Italia, Polandia, Filipina, Taiwan, Turki, dan Cina. Pembelajaran di Universitas Sriwijaya dikontrol melalui mekanisme untuk menjamin kelangsungan perguruan tinggi berkualitas dengan fasilitas penunjang yang representatif, perpustakaan, masjid, klinik, student center, apartemen mahasiswa, kantin, auditorium, dan sarana olahraga.
Ladies and gentlemen, let's continue to the next agenda, plenary session. The chair of this session is Dr. Fitri Maya Puspita. To Dr. Fitri Maya Puspita, time and place are yours.
Thank you. Ladies and gentlemen, good morning. Welcome to Sriwijaya International Conference on Basic and Applied Sciences, hosting by Faculty of Mathematics and Natural Sciences, Sriwijaya University. First, let me introduce myself. My name is Dr. Fitri Maya Puspita. I'll be responsible for hosting this presentation today, and I'm glad to welcome Professor Marcelo Orlandi, Professor Dr. Puji Lukitowati Haryani, Associate Professor Dr. Walid Fuad, Professor Fadzila Adibah binti Abdul Majid, and Associate Professor Arif Gusnanto, PhD. There are five keynote speakers here. Before starting the presentation, let me explain one by one his or her brief curriculum vitae first. For first presenter would be Professor Marcelo Orlandi. His primary position as a Vice Director of the Innovation Agency and Associate Professor. His country from Brazil, from the Sao Paulo University. His professional employment recently would be the Vice Director of UNESP Innovation Agency, Sao Paulo State University in Brazil. His education would be in 2005 until 2008 a PhD degree in Materials Science and Engineering of Federal University of São Carlos, UFS, CAR, Brazil. His interest would be in synthesis of inorganic materials using vapor phase, solid background in both scanning and transmission electron microscopy, SEM and TEM, analysis of electrical properties and gas sensor application of semiconducting metal oxides. He is a coordinator of electron microscopy facilities at Chemistry Institute, UNESP. This facility includes one SEM FEG and one TEM and one FIB dual beam microscope. So, ladies and gentlemen, without further ado, please welcome Professor Marcelo Orlandi, who will present his topics on the use of tin oxide materials to detect air pollution. Time is yours, Professor. Thank you.
Hello, thank you everybody for this very nice introduction. So it is my pleasure to be giving this talk at SICBAS. And for me here in Brazil, it's still November 1st. I know for you it's November 2nd. And today I'll talk about a little bit my research interest that is related to gas sensing application of metal oxides. I will focus on tin oxide. So let me share my screen. I hope all of you are seeing my screen. Is it?
Yes.
Are you?
Yes.
Okay. So I'll talk about the tin oxide for gas sensing. So I'm not working just with tin oxide, but because we need to focus. Today I choose to talk about it. I like a lot this material. I'm not going to talk just about SNO2, the most famous material about tin oxide, but also other phases like SNO and SN304. So it is really important for us to have the emissions control. As you should know, we have this problem, and most of the pollution we have nowadays we are having this week in Glasgow, some people trying to solve the pollution on the earth, and it is mainly from chemical industries, power plants, and automobiles. So here we have some pollutants and their maximum value that you can have for 40 hours per week workers or short exposition limit. And as you can see here for NO2, NO2 is very critical pollutant, so the levels that it should be detected is very low in ppm levels. And this is very interesting slide about the China before the lockdown and after the lockdown, the levels of NO2, some cities. So we can see what it's showing us, that decreasing the industrial activity, it is possible to solve the planet. So it's in our hands to do it. It's on the governments to do something, trying to have a healthier planet. And today I'm going to talk about these very interesting semiconducting metal oxides. I like a lot this kind of materials. And here we can see on the left that if you have the material in ultra high vacuum, we are going to have a flat band, and on exposition to air, we will have some oxygen adsorbed on the surface of material, and in this case, some of the electrons of material are going to be trapped, and it is going to have the band bending on the material. And with this kind of band bending, if it is going to increase, we have a change in the material resistance. So if for a typical N-type semiconductor, the resistance is going to increase. And we can also change the material, putting another material like here, water vapor. And in this case, it can be reducing material or even hydrogen. And we are going to have an increase in a decrease in the resistance of material. And having another oxidant materials, we can have the resistance modulated again. So it is possible with the analytes that we have on the atmosphere, have the control of the resistance. And on the other hand, in this case, what we're going to do is be analyzing all the time the resistance of the material and then try to detect what are the analytes and quantify it on the atmosphere. We can do it just with one sensor, like here, SNO2, and we can do it also on an array. And this is a slide from Professor Harry Tuller from MIT that we have a close cooperation since 2010. And this is the principle of working of these semiconductors. So when you have the surface of a typical N-type semiconductor like NO2, SNO2, and we have oxygen adsorbed on the surface, we are going to have electrons trapped. And here we have a depletion layer, a layer that is depleted of electrons. And if this layer is depleted of electrons, for sure, the resistance of material is going to increase. And it means that we have a potential barrier at the grand boundary of the material. If we introduce some reducing gas, like CO or hydrogen, so what is going to happen is the CO is reacting with the adsorbed oxygen, and in this case, electron is going to be released to the bulk material, and the potential barrier is going to decrease, and the conductivity of material is going to increase. So Depending on what kind of analyte we have on the surface, if it's oxidizing, if it's reducing, we have the modulation of the electrical property of material. So, like I told you before, I'm going to focus on tin oxide. Tin oxide is a very good material to study the gas sensing properties. So SNO2 is a standard material for gas sensing application. Most of applications is related with SNO2, but it's also possible to control the phase, not only the famous SNO2, but also the SN3O4 and the SNO, if you have the control about the oxygen during the synthesis materials. And it's really interesting that there are few studies about SN3O4 and SNO. And for us Brazilians, it is really important since we have 6% of the global reserves of cassiterite. And recently we, together with Elsevier, we launched this tin oxide book, and it has more than 600 pages just focused on the basic properties and applications of tin oxide. So going to the results, we can have after the synthesis, and as it was told in the introduction, the synthesis can be by vapor phase. We can have these white, yellow, and brown material. And doing the electron microscopy analysis, we can see like 1D material for the white and yellow one, and the disk and the 1D material for the dark one. So we went to analyze this material deeply, and we realized that the white one is SNO2 as expected, but since it is 1D material, it has a preferential orientation for the 101 direction, that is the direction of these nanobelts. And for the yellow material, the main phase is the SN3O4. And it's really interesting that these materials, SN3O4, they are layered. And we recently showed that these layers, they are glued together by van der Waals force. So here we have an ectaxial growth of the layers, and here on the SEM images, we can see that the material is really layered. About the dark material, as I showed before, we have two kinds of morphologies. We have also the nanobelts, and these nanobelts have this exposed phase. The most, the largest one is the 001. And we also have the discs, the SNO discs. And it's interesting that this higher surface of the disc, this is like a 2D material. It's also the 001 surface. And please keep in mind, because our results is going to be important, this exposed surface of the material 001. So this is some sensor response of the materials. And as I told you, the SNO2 material is shown in blue, is the standard one, and the response is very nice. So we can have here 100 sensor response for 100 NO2 exposition. But if you take the SNO micro discs, it is 1000 change in the resistance. So this is what we call the giant chemo resistance response of this material, because it's much higher than the standard material that we usually have for the NO2 gas sensing. And in this case, we can easily detect 0.1 ppm. And if you remember at the beginning of the talk, we need to detect about 4 ppm in this case. So it easily can be used in practical applications. So it is very common, people say about the selectivity and also the stability of material. Why? Because at 400 degrees, we have the disproportionation of the SNO becoming SNO2 plus metallic tin. So it is really important that the best sensor response of this material is below 400 degrees. And in this case, we can see that the best sensor response is at 200. So the material is pretty stable at 200 degrees. And another important thing is to have the surface decoration of the material. And here we also studied the nanobelts and the discs with the decoration of noble metal nanoparticles. And we can have two kinds of effects on the gas sensing response. That is electronic sensitization. It's related to the Fermi level of the nanoparticle, related to the Fermi level of the bulk material. And we can also have some chemical sensitization when the nanoparticle is acting as a catalyst. And for the case of the discs, we studied palladium and silver. And as we can see here, with the pristine one, for NO2, the pristine is much better than the decorated materials. But if we try to detect hydrogen, so what we can see here is that palladium is much better than the pristine and the silver one. So in this case, we have exactly the same material, but with surface engineer, some surface functionalization. And in this case, we can change the selectivity of a material. In this case, this change in the selectivity is related to the catalyst effect of palladium. So first we have the hydrogen molecule on the surface through the split over process. And after we have the spillover process when this hydrogen is going to the material surface, and we have these extra electrons going back to the bulk of material, and in this case, increasing the conductance. And it works not only with the nanodiscs, the microdiscs, but also with the nanobelts. So for the nanobelts, we also tried the palladium and the platinum. And we can see here that the pristine one for NO2, always the pristine one is the better sensor response. But if you are trying to detect hydrogen, the material, the system with palladium is the best one for the same reason that I showed before. And in this case, we can see, as I showed you, the electronic sensitization effect about when we have a platinum together with SNO, the mismatch between the Fermi level is higher. So in this case, we have a strong electronic sensitization. And with the palladium, we have metallic palladium have a lower electronic sensitization, but we can have it higher as the palladium is going to the oxide. And during the exposition and reduction of material to the analyte, the palladium can be oxidized and reduced. It is very common for this kind of material. And finally, I would like to show you about this dual beam nano fabrication, and it's possible to do single element sensors. I'm not going to enter in too much details about the dual beam, but basically we have two beams, one of ions and another of electrons. And we did some nano devices and nano discs for just one single element for gas sensing application. The main importance of it is the near zero power consumption as we are talking. So it's really important to save energy. And we can see here as a proof of concept that these nano devices can also work. For sure, the sensor response is not so high as in the carpet mode material because of the grand bonder is absent here. but it can also work as a sensor. So here is the summarizing the results. We can have SNO, SN304, and SNO2 nanostructures, and they were synthesized by carbothermal reduction method. We can have the SNO discs presenting giant chemo resistance response, and we can have the change in the selectivity of a material due to surface degradation. And it's really important to know about the Fermi level of nanoparticles because of electronic sensitization. And if we can have a nanofabrication, it is possible to have a gas sensor device working and with near zero power consumption. So I would like to thank all the funding agencies that is paying our bills here, and also would like to thank the organizer of this very nice conference. And I for sure would like to thank all the students and the collaborators, and I'll be glad if you have any question. So thank you very much.
Thank you very much for Professor Marcelo Ornagi Orlandi. We move to second speaker would be Professor Dr. Puji Lukitowati Haryani. She is from Indonesia. The affiliation is from Department of Chemistry, Faculty of Mathematics and Natural Sciences, Universitas Sriwijaya. Her education is PhD in Environmental Science, Universitas Sriwijaya, graduated in 2013. Her research interest is focused on nanomaterials, especially for nanomagnetic and nanomagnetic modification for environment. Her publication recently includes Scopus for 36 articles, non-Scopus for 127 articles, 2 books, 2 granted patent, and 5 patent registered. Her presentation title would be The Application of Magnetic Photocatalyst for Ferrite Compound Modification for Water Treatment. So with no further ado, please welcome Professor Dr. Puji Lukitowati Haryani. Thank you.
Thank you for moderator for the time given. Good morning, all participants of the Sriwijaya International Conference on Basic and Applied Science. Assalamualaikum warahmatullahi wabarakatuh. I'm from Department of Chemistry, Faculty of Mathematics Natural Science, Universitas Sriwijaya, and Research Center of Advanced Material and Nanocomposite, Faculty of Mathematics and Natural Science, Universitas Sriwijaya.
Professor, could you give a slide show?
Okay, thank you. My research focus on nanomagnetic and modification of nanomagnetic composite for environment. The title in this presentation, Application of Magnetic Photocatalyst ferrite compound modification by silicon oxide, metal oxide for water treatment. My topic is similar Prof. Marcelo about my research focus of material for water. Water pollution is one of the most egregious environmental problems that have caught the attention of researchers worldwide. The origin of this problem is rapidly increasing industrialization and population expansion, which contributed to water pollution. The direct disposal of untreated sanitary waste, toxic industrial effluents and runoff from agricultural fields into rivers and water resources are the primary causes. The majority of the world population encounter the problem of clean drinking water. And the condition are advanced in developing countries. The outbreak of waterborne diseases like typhoid, diarrhea, cholera is the major cause of infant mortality. Suitable water sources are required for human consumption, industry, agriculture, and recreation. The increased water usage will ultimately generate less influence of wastewater. Although more than 70% of the earth is covered with water, but only 3% is suitable for human consumption, and the remaining 97% is salty water. Around 4 billion people worldwide face water scarcity for at least 1 month annually. So clean water is essential element for use. WHO reported 2025 about 50% of the world inhabitants will be living in water stress area, and until 2015 only around 2% of the contaminated water was properly treated over the world. So in view of this peril effect, there is an urgent need to affordable and reliable water purification technology. Several techniques have been reported for wastewater treatment. They typically include physical, chemical, and biological process considered effective enough for water treatment in many ways. Their selection depends on various factors like pollution concentration, sewage composition, cost of the process, and the additional impurity present in wastewater. The distinct feature of its treatment method can be beneficial in one way, but restrict in another. Among physical method, absorption has been used and by research in recent years because of its high efficiency and low operational cost. The most commonly used conventional absorbent for pollutant and dye elimination are bioabsorbent, silica, alumina, activated carbon, clay, metal oxide, titania, and so on. Several chemical oxidation process are reported for a range catalyst application. The advanced oxidation process is considered an essential technique for wastewater. Advanced oxidation process is used wastewater treatment that have commonly principle in generating oxidizing species such as hydroxyl radical. Microorganism becomes or degrades organic colorant in biological treatment via aerobic or anaerobic cycle. Microbes use organic as an energy source by degrading them. The biological method for the complete degradation, for example, textile wastewater have benefits such as being eco-friendly, cost competitive, less sludge production. Therefore, many microorganisms and enzyme has been isolated and tried to for the degradation of several pollutants. A wide range of microorganisms such as bacteria, fungi, algae can degrade a wide variety of dyes present in wastewater. The public will accept that a biological treatment is a simple and economically attractive technique, but it is slow process and require an optimally favorable environment and proper maintenance of microorganisms. Why use nanomagnetic for treatment of wastewater? Nanomagnetic have been have number of physical chemical purposes that make them particularly attractive for wastewater because nanomagnetic can be removal in organic and organic pollutant. High absorption of hazardous pollutant, reduction the toxicity of different pollutants, providing modifiable reactive surface to increase the capacity absorption, cost effective and reusability. And the absorbent can be separated easily and quickly after the absorption or photocatalytic process using an external magnet. Nanotechnology is considered emerging and cutting-edge technology with a wide range of application in different fields of science. In past few decades, there has been increased research interesting in forming a novel nanomaterial, particularly for biomedical and environmental protection. It can be offset from the published literature that nanoparticles have been used for the removal of heavy metal, toxic dye, halogenic compound, pesticides, pharmaceutical, and personal care products from industrial effluents. Magnetic nanoparticle research is growing rapidly and evidenced by the many publications as shown in the slides. Modification nanomagnetic is very necessary to stabilize the nanomagnetic because nanomagnetic easy to agglomerate. The nanomagnetics, their small size, easily oxidized so that the solve is in solution. Various type compound can be used for nanomagnetic coating, such as using polymer, for example, polyethylene glycol, chitosan, EDTE, and so on. For example, nanomagnetic stabilization using natural ingredient. So in the slide, using crude extract of Garcinia mangosteen fruit peel. The crude extract contains several groups of organic compounds, such as flavonoid, anthocyanin. My research and modification, nanomagnetic iron oxide with hydroxyapatite to remove the heavy metal and phenol. The absorption capacity of hydroxyapatite is more than hydroxyapatite PEG iron oxide for absorption phenol. So that this provides the coating increasing the surface area and absorption capacity. Activated carbon iron oxide for absorption process red dye from wastewater songket industry. From the slide, we can see that significant color change before and after the addition of activated carbon magnetic. Activated carbon iron oxides has efficiency of reducing has 19.4% and reducing the chemical oxygen demand until 96%. For my research, so that the toxicity test using tilapia first, also so there was water songket industry after being processed did not cause the tilapia test animal. Modification nanomagnetic ferrite compound with silicon oxide and titanium oxide. Ferrite compound, for example, iron oxide, copper ferrite, cobalt ferrite as and silicon silicon oxide, and semiconductor, titanium oxide, nickel oxide, and so on as a core. The effect of the modification, magnetic core fall separation in magnetic field, and interlayer preventing photodissolution and chair carrier recombination, and reduce the band gap semiconductor so that it can be used for the visible region, not only in ultraviolet region. In my research, nickel ferrite nanomagnetic as photocatalyst for conglomerate dia. The mechanism of photocatalytic degradation generally involves the formation of hydroxyl radical and the formation of superoxide anion from oxygen. When a photocatalyst compound absorbs light with a specific wavelength, the catalyst obtains energy. This energy is used for the excitation electron from the valence band to the conduction bands. The resulting Hall breaks down water molecule into a hydroxyl radical with reacts with organic molecule to break down organic compound. The slide shows GCMS analysis of conglomerate after photodegradation. Conglomerate dia is degraded into small molecule. And the final product is water and carbon dioxide. If compared to the absorption method, this method is better because it does not cause secondary pollutant. The mechanism of photodegradation similar with previous mechanism. Schematic of photocatalytic degradation process, the pollutant mixed with catalyst, followed by absorption process, and then photocatalytic degradation process with the help of energy lights. After degradation, the catalyst can be separated from the solution using magnet external and reused for the photocatalytic degradation process again. From the ratio scanning transmission electron, we can see that iron oxide nanomagnetic as a core and silicon oxide and titanium oxide as a cell. Titanium oxide is evenly distributed. This is important for the photocatalytic degradation process to run effectively. From my research, saw that the coating nanomagnetic with the other compound decrease in saturation magnetization, but nanomagnetic ferrite compound modification still had strong magnetic properties. Photodegradation of methyl orange using modification cobalt ferrite compound with silicon oxide and titanium oxide have to maximum wavelength in the UV and visible region. This advantage of using titanium oxide is only in the UV region. This phenomenon is supported by a measurement result of band gap, where the band gap of titanium oxide of 3.4 after coating becomes to 0.5 electron volt. From the transmission electron microscope, cobalt is heterogeneous particle. Coating with silicon oxides show a large size, and also coating with titanium oxides appear to have a large size and coagulation. Several factors affect photocatalytic degradation, such as pH solution, initial concentration dia, and irradiation time. The photodegradation of methyl orange using cobalt ferrite modification so that has adequate linearity and follow the Langmuir-Heisenberg model expressed in pseudo-first order. The regeneration and reusability of catalysts are highly significant in industrial because this property are related to cause, payload scale remediation system, and environmental safety. In my research, the catalyst effectiveness reduced about 3.74% after full cycle, yeah. And then the catalyst can use to photocatalytic degradation again. In general, wastewater contain pollutant as well as pathogenic bacteria. Nanomagnetic has the ability to inhibit the growth of bacteria. In my research, iron oxide coating with PEG so that iron oxides have antibacterial activity to the Staphylococcus aureus and Escherichia coli. From the same image, bacteria treatment with nanomagnetics, the bacterial cell wall actually leads to a breakage of the structure. Slide antimicrobial activity cobalt virus for MIC value, IC50 and MBC for bacteria S. coli, Escherichia coli, Staphylococcus aureus, and so on. Nanomagnetic and modification of nanomagnetic can be synthesis with physical methods, chemical method, and biological methods. In biological synthesis, plant extract contain different metabolites like terpenoids, phenol, protein, carbohydrate, which are directly responsibility to reduce the metal 1 nanoparticle and also escaping stabilizing again by hampering to aggregation of nanoparticle. Co-precipitation, salt gel, hydrothermal, sulfothermal, I commonly use method for nanomagnetic synthesis. The process influenced by the precursor, the concentration of sodium hydroxide, stirring, temperature, and so on. Solution combustion is synthesis using organic compound as a fuel. The synthesis process is fast and requires high temperature. Different fuel products. Okay, one minute again. Okay, 1 minute again. Product different nanomagnetic characteristics such as size, magnetic properties, surface area. In my research, synthesis of zinc ferrite using EDTA have a large saturation magnetization. The conclusion, many techniques have been explored exhibiting their superlative application in removing the pollutant from wastewater. Modification of ferrite compound using silicon oxides, metal oxide as a superconductor is effectively used for catalytic degradation of pollutant in the small and harmless molecule. The catalyst can be separated from solution quickly using a permanent magnet and can be reused for catalytic degradation again. And various methods can be used for nanomagnetic synthesis and modification. Thank you, moderator.
Give applause to Professor Puji Lukitohati Haryani. Okay, we move on to next speaker would be the Associate Professor Walid Fuad, PhD, with his presentation title is The Road to Develop an Efficient Sweet Sorghum Crop for Biofuel Production. Before we listen to his presentation, let me give brief curriculum vitae of Professor Walid Fuad. He is Associate Professor at Plant Biotechnology at the Biology Department of and Biotechnology Program at the AUC School of Science and Engineering. His research interests focus on the integration of genetic transformation technology and functional genomics to study, identify, isolate, and engineer key factors for molecular improvement of crops in general and biomass crops in particular. His goal is to initiate and develop products that will improve agricultural production at the regional and international level. He is interested also in the use of biochemical and molecular biology tools to understand and improve environmental and biotic stress tolerance in plants to enhance their productivity and persistence, resulting in more efficient use of natural resources. His education in 2004, he pursued his PhD from University of Florida, Gainesville, Florida, United States of America. With no further ado, let us welcome Associate Professor Walid Fuad. For his title of presentation is The Road to Develop an Efficient Sweet Sorghum Crop for Biofuel Production. Thank you.
Thank you so much, Dr. Fitri, and for the introduction. And I would like to thank you also the organizing committee for the invitation to this meeting. I think I'm sharing my screen now, right? You see the full presentation? All right. So as was introduced, the title of my talk today is The Road to Develop an Efficient Sweet Sorghum Crop for Biofuel. So before I go into detail about my presentation, I'll give you a small introduction about sorghum and what are the different varieties of sorghum. And I will give you also a brief introduction about why am I interested in biofuel, and how biofuel can be produced, what is about bioethanol production right now and worldwide, and why there is a need to switch from the current practice of bioethanol production to a much more sustainable approach. Then I will give maybe talk about two aspects of my ongoing research, which is kind of going hand by hand, which is number one is evaluating current germplasm for their potential to be used as a biofuel, and how can and the plan to improve, or developing a tool to improve the current germplasm. So sorghum, this is a picture for the sorghum growing at in Cairo at the American University in Cairo campus, and sorghum originated mainly in Africa, and it's close relative to sugarcane and corn. Sorghum is widely used, and it's been kind of it's called like an ancient crop. It's been used, and it's still currently used as a main food or grain crop feed, grain food for different tribes and in African countries. Worldwide, sorghum ranks 5th after the 4 major grain crops, including wheat, rice, corn, and barley. It's, when you compare it to its relative like sugarcane, sugarcane takes almost like 12 months for maturation and harvesting. Sugar, on the other hand, sorghum stay only for a short period of time, and it's harvested within maybe 4 to 6 months based on the variety and the application. Sorghum also, it's a C4 plant, and C4 plants have evolved a special technique to improve their water content and kind of avoid stresses during high temperature stresses, and or during high temperature in general. or with the low water availability in the soil. Therefore, C4 plants tend to be much more efficient in growing in arid and semi-arid area worldwide. Sorghum is quite diverse genus, so there are it's one among the very diverse genuses. So there are the most popular sorghum is mainly the green sorghum, which is grown for the seeds, and there are also sorghum that grow that cultivated mainly as for forage. So in this case, you'd like the entire yield of the plant is being used for as an animal feed. And there are also sweet sorghum varieties, and the sweet sorghum variety is known to or characterized by accumulating high sugar content in their stocks, and they can accumulate up to 20% sugar in their juice, which is quite comparable to the sugar cane. So they are very efficient and a good source for sugar. So what makes sorghum interesting crop as well is the grain is gluten-free, so it does not have gluten, and it's right now there's like a shift toward gluten-free food. Therefore, it is one of the best candidate foods to source of energy compared to wheat, for example. Sweet sorghum, as I mentioned, is used for syrup production. So it's used like for widely used in the US. It's used for animal feed, and what the focus of our talk today is mainly bioethanol, or maybe that's like one of the main reasons. So ethanol production from sorghum can be obtained from different sources. So if you go back in history, the idea of utilizing ethanol as a biofuel is really as old as Ford established his Model T engine. And in the 1930s, corn was actually considered in the US as the main source or proposed to be a kind of a source for alcohol to run gasoline. And this movement was kind of taken over by the discovery and the wide production of fossil fuel. Therefore, it was not really an attractive approach until the oil prices reached very high prices. And for example, in the 70s, there was a significant increase in the oil production. So in the oil prices, therefore, some countries, specifically Brazil, switched their most of their cars to run on ethanol. But the ethanol production in Brazil is mainly utilizing sugarcane. In 2004, the US also switched to corn as a source of ethanol. So they kind of revived this old movement because of the soaring oil prices. And they started to produce ethanol mainly from corn. So if you look at ethanol production worldwide right now, you see the largest producer of ethanol is mainly the US. So the US almost produced more than 50% of the global ethanol production. The vast majority of this is coming from corn ethanol. The second largest producer is Brazil. And Brazil also produces ethanol from sugarcane, as I mentioned earlier. So both together, they kind of dominate more than 80% of the world production of ethanol. As you can see, using corn or sugarcane as a source of ethanol is actually competing with our food. So both of them, sugarcane and corn, are actually stable crops for human food. And switching to use this type of crops resulted in a significant increase in the corn prices, almost doubled when the US switched to the production of ethanol from corn. Almost 40% of the corn produced in the US currently are utilized for corn production, for ethanol production. So it's very you can kind of the importance of switching to ethanol is mainly the kind of declining production of oil, the increasing prices of oil, fossil fuel, I mean, and also the negative impact of the utilization of fossil fuel, as it's actually add or increase the carbon dioxide in the atmosphere. Utilizing on energy from plants, where the plants actually fix this carbon dioxide, so kind of make the carbon actually recycled between plants and the atmosphere, even though we're using it for energy production. So because of the soaring prices of corn and sugar cane, there was a significant demand for switching to other plant material as a source for biofuel. And if you look at the plant composition, plant cell wall is composed of polymers of cellulose and hemicellulose. Both of cellulose and hemicellulose are actually polymers of glucose and xylose, respectively. And these fibers are kind of supported by another component, which is a lignin. So lignin works like a glue to kind of stabilize the cell wall composition. So this cell wall composition can be broken down and hydrolyzed by enzymes to produce monosaccharides, either 5-carbon sugar or 5 or 6-carbon sugar. Then this 5 and 6-carbon sugar can be fermented to produce ethanol. So this is kind of the ideal situation to fade away from the current utilization of food as a source of ethanol. When you look closely at the cell wall composition, you can see that it's only the cellulose and the hemicellulose that actually contribute to the ethanol production. On the other hand, lignin kind of hinder ethanol production, and specifically hinder the enzymatic hydrolysis of these fibers. And there was a lot of studies that were conducted that say that the very strong negative correlation between the amount of sugar released and the lignin present in the cell wall. So the more lignin you have, the less sugar can be released from the cell wall fraction. Therefore, my main focus on the objective today is mainly focusing on kind of screening different sorghum inbred lines for using for different traits. Phenotypic traits in terms of the yield and the production, and the biochemical traits, which is mostly the cell wall composition, which looking at the cellulose, hemicellulose, and the efficiency of the breakdown of this cell wall composition to liberate monosaccharides. The second objective, which is we managed to identify a potential candidate or for improvement. How are we going to improve it? To do the improvement. And the second part of the talk is mostly focusing on developing a system that allow us to improve sorghum quality to fulfill our needs. So this is this picture. So starting with the first objective, we have collected, we screened different varieties that growing here in Egypt, and we selected 7 of these varieties, and we grow them at different time in the field using drip irrigation. You can see here, and this picture shows the plants at 21 age, 21 days after sowing, after planting, and this one at 60 days after planting. So these plants were harvested at 2 time points mainly, and were also scored for different phenotypic parameters, including the plant height, the number of leaves, the fresh weight and the dry weight, the dry matter, so on. And it was also analyzed for the cell wall composition, the different fractions of fibers in the neutral detergent fiber, acid detergent fiber, and lignin, and crude protein, and so on. So looking into the results, we can see that there are some variability among the different species when it comes to their heights, and also when it comes to the stem diameter. So the higher the plant, the thinner the diameter is. So the shorter the plants tend to be much more efficient in terms of accumulating biomass and avoiding lodging and a loss of the biomass. So we can see here at least these 3 varieties, Ramada, Rex, and Sugar Drip, having higher superior biomass and superior diameters and reasonable height as well. Looking at the number of leaves, which is an indication for how much biomass you have you can see that these three varieties again, along with this MON 1054, they have the highest number of leaves per plant, and the leaf width is also give you an indication about how much tissue do we have, and we have again the Ramada sugar dip having the highest the leaf width. Looking at the fresh and dry weight, you can see the Ramada is having the highest fresh weight and highest dry weight as well when we look at the along with sugar driven mix. So we concluded that these three varieties are actually could be a potential target for improvement. We wanted to kind of examine their biochemical properties as well. This table, I mean, avoid my too much information here, but I highlighted certain parameters. This is literally the analysis of the fiber composition within the plant of the cell wall fraction. As I mentioned, the NDF, the ADF, the ADL, all of this are actually parameters indicating that the lower the NDF, the ADF, and the ADL, the highest the efficiency or the highest the potential for utilization of these fibers for releasing elements, releasing the monosaccharides. And as we saw here, the lignin is one of the main important things we were looking at, and we can see lignin content is the lowest in one of the varieties that showed superior phenotypic traits. This is only the fractions, so we took this fraction and we started to apply some sort of like much more in-depth analysis, which is the breakdown of these fibers. So we run in vitro digestibility for the dry matter and in vitro digestibility for the organic matter. And we can say here that this variety, along with the Rex and the sugar drip, have the highest in vitro digestibility, means that they will release more monosaccharides compared to the other varieties. And that's definitely might be related to the availability or the low lignin content that they have. So we concluded from the study that these three varieties have the very good potential to be improved. Therefore, we move to the next objective, which is establishment of an efficient in vitro and transformation system for this suite of varieties. So why do we need to do this transformation system? Because yeah, even though it has a sorghum as a high potential to an energy crop, but all crops have some sort of issues when it comes to, or problems when it comes to their growth. For example, diseases and being susceptible to diseases or insects are some of the drawback for sorghum. And maybe we can improve the sugar content or improve the biomass of the sorghum itself. So we can do this also by utilizing molecular techniques, or maybe improve its tolerance to environmental stresses. So we can engineer the plants to gain more benefit from it by make it more resistant to insects or so on. But why we told this route? Classically, if you want to introduce new traits from one plant to another, you do what we call the classical breeding. And the classical breeding is literally kind of elect some donors and cross these two plants together and start selecting for the best traits. So this, even though it's successful and it's been mainly the main approach for the production of one of the varieties that we have right now, but it takes quite a long time. And sometimes the traits that you are looking for is not available within the germ plants. So therefore, it will become difficult to transform certain traits. On the other hand, if we use the molecular biology technique, you can specifically take certain genes of interest from whatever source, whether they are within the germplasm or outside of the germplasm, and engineer this genes into the traits that you are, the variety that you're interested in. And usually this takes a little bit much, almost like half the time it takes for the classical breeding. So we're applying this approach or utilizing this approach in my lab. So let me tell you, how can we introduce genes into the plants? The most important thing is developing what we call in vitro regeneration system. And using the in vitro regeneration system, what we do is you take any part of the plant and grow it under different types of plant growth regulators and make this tissue that you excise or the explant regenerate into a whole plant. During this phase, you can introduce whatever genes that you are interested in, and these genes can be transferred to the entire plant, and the entire plant become transgenic and harboring the gene of interest. So in order for us to achieve the efficient regeneration system, we utilize PGR, different PGR.
Excuse me, Professor, sorry for 2 more
Yes.
minutes, please. 2 more minutes.
All right, thank you.
Okay, thank you.
All right. So we utilize different types of PGRs, and we can also utilize different type of explants. So we can utilize immature inflorescence, leaves, immature embryos, or so on. The approach that we took is mainly utilizing immature inflorescence. So we harvest immature inflorescence at different phases, and we cut them into circles or discs, and we grow them under different media composition, and we look for this mass of undifferentiated colors to grow. And then we move this to another type or another combination of PGR and make it originate into plants. So I go quickly here. I don't want to stop much. So you can see here, this is the results of examining different media composition, and you can see a very efficient production of what we call undifferentiated mass that will be later on developed into whole plants. We take these plants and move them to different media, and this media can generate a whole plantlet. Again, we managed to have very good efficiency when it comes to specifying different types of PGR to regenerate the whole plants in vitro. So we established a system. We can go from immature inflorescence to a whole plant. This is growing in the greenhouse. Now the next step is literally doing the transformation. So we used one varieties was mainly sugar drip, and we utilized the optimized system for the regeneration. We initiated the culus, and at certain phase we introduced the foreign DNA that would be inserted into the plant, and we grow the callus, and we did the selection, and only the plants that have the genes that was introduced into this plant were regenerated and developed the healthy plants. So from these two experiments that we conducted, we managed to develop almost 1.7% efficiency for the transformation system in the sugar drip. Therefore, to wrap my presentation, we can say that we managed to evaluate kind of select the best prospective line suitable for biofuel production based on the cell wall composition. We also utilized we developed a very efficient system of the in vitro regeneration and transformation that will facilitate or allow us to do further improvements. What are we doing right now in my lab is literally we are targeting lignin composition. So we're targeting lignin by synthetic genes by two techniques, one of the genome editing technique and the RNA interference, and this will reduce the total lignin content in the plant and make them more suitable for biofuel production. We're planning also to improve the transmission efficiency by manipulating some factors. We are also introducing some genes involved in the plant responses to oxidative stress to make them much more tolerant to environmental stresses. And lastly, we are trying to improve the biomass by altering the plant architecture. So all of this work is going on, is ongoing projects right now in my lab. And to finish, I would like to acknowledge my collaborator, Dr. Sreen Asim from Agricultural Research Center, and the 3 graduate students who were involved in this project, and the Center of Applied Research and Environmental Sustainability at AUC for growing the plants. And definitely all this research was funded by the AUC, the American University in Cairo Internal Research Support Grant. And thank you again for the chances to share my ongoing research with you, and I will be looking forward for discussion after at the end of the panel.
Thank you, Associate Professor Fuad Walid. So, ladies and gentlemen, we move on to the 4th keynote speakers, would be Professor Fadzila Adibah binti Abdul Majid. She is from Department of Bioprocess Engineering, Faculty of Chemical Engineering, University Teknologi Malaysia, Johor. Before going to her presentation, let me give brief curriculum vitae of Professor Fadzila. In 2002, she pursued her PhD in chemical engineering in the University of Birmingham, United Kingdom. For her recent publication would be the quantification of gallic acid and tannic acid from Quercus infectoria or manjakani and their effects on antioxidant and antibacterial activities, and also the total phenolic content and antioxidant activity of Quercus infectoria galls using supercritical carbon dioxide extraction technique and in comparison with chocolate extraction. So, with no further ado, let us welcome Professor Dr. Fadzila Adibah Abdul Majid. Thank you.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi ta'ala wabarakatuh. Thank you, moderator, and thank you for the brief introduction of myself. I'm so sorry that I would like to make some clarification. That is actually I am now working in the University Malaysia Terengganu. I was the lecturer in University Technology Malaysia for the past 23 years, but I have been in University Malaysia Terengganu for the past 6 years. Okay, so I am from Marine Institute Biotechnology, and today I'm going to talk about the marine discovery work. Okay, so I would like to share my presentation. Okay, so the topic of my presentation is marine biotechnology for the drug discovery and beyond. So, as a University of, before I start, I would like to say thank you to the organizing committee for inviting me to this auspicious event, SICBAS 21, and my title is Marine Biotechnology for Drug Discovery and Beyond. Okay, it is interesting that our ocean representing 70% of the Earth, and we are sitting on the only 30% of the land available on this Earth. So there are a lot of opportunities to be discovered under the water. Okay, so I would like to share these slides. You see that previously in the not really ancient time, but previously we did Malaysia, Indonesia, and this region is called Nusantara. We are actually a nation, one nation that claim the land and the sea are our territory. So this is a very interesting to note that we call our the place that we live as tanah airku, so which is the land and the water. Okay, so actually there are a lot of beautiful things under the water of this tropical area of the Nusantara. Okay, so the salt water has enormous marine resources that is not yet being discovered. So we hope that this area one day will be the main resources of our future products, be it the chemical, be it the food, the future food, and the future medicine, and so on. So actually, underneath our water, so there are so beautiful. Okay, so of course that today the current activities related to the ocean, like oil and gas, like fisheries for food, for tourism, transport and logistics, are the important businesses that contributing to the enormous amount of economic values to countries, okay, especially the countries around Southeast Asia region. Okay, but what lies underneath is a lot more than we can actually discover if we know how. Yeah. So with this, University Malaysia Terengganu has been established to focus their activities related to ocean. So our university has the motto of Ocean of Discoveries for Global Sustainability. So our main focus are related to marine activities. So here we are, University Malaysia Terengganu, situated in the east coast of Peninsular Malaysia. We are quite a small university, but our university is very situated in the near the sea. So it is like a hotel. Okay, so our faculties are Faculty of Marine Studies, Faculty of Science and Marine Environment, Faculty of Ocean Engineering, Technology and Informatics, Faculty of Fisheries and Food Science, and also Faculty of Business, Economics and Social Development. And we are also supported with one Center for Foundation and Continuing Education, and also STEM Foundation. Apart from that, we also have 4 institutes that mainly focus on the marine studies. We have Institute of Oceanography and Environment. We have Tropical Aquaculture and Fisheries. We have Marine Biotechnology, and also Institute of Tropical Biodiversity and Sustainable Development. So what I'm going to talk today in a big length is what are the activities of Institute Marine Biotechnology with regards to the drug discovery. Okay, so this is our Institute of Marine Biotechnology. The vision is to be a reputable research institute and reference center for marine biotechnology at the national level and respected globally. So our mission is to spearhead the exploration of new resources through sustainable management by merging expertise and advanced biotechnology application. So when we establish our institute, we chart our pathways from doing the fundamental research up to the drug discovery, up to the development of novel drug from marine resources. So these are the pipeline of our work. It's a bit lengthy, but following this, we are on the right track. Yeah. So in order to make a new discovery in drug from marine resources, we established several laboratories in our institute. We have natural product and synthetic laboratories, microbiology lab, we have plant cell culture lab, we have bioinformatics lab, bioprocessing lab, immunology lab, animal cell culture, genomic lab, proteomic lab, and also animal house. And these, and currently, recently, very, very recent, we add another laboratory which cater for climate adaptation research work. So with these facilities, we are ready to start our research work. Yeah, okay. So in order to speed up our research, we divided our research activity into 4 programs, which are a natural and synthetic product program. We have molecular and immune and molecular therapeutic program. We have marine microbes and environmental program, as well as bioinformatics. So these all 4 programs have their specific task in order for us to achieve the target of developing a drug from marine resources. So before we go on, it is very interesting for me to share this data. This map showing Ocean Biodiversity Index. If you can see, biodiversity index can be from 0 to 1, yeah, and then it is very interesting to see that the ocean around Southeast Asia region is considered to be very fertile and very lucrative with marine species. So we have the global marine hotspot based on biodiversity index using the scale showing that this area contains more than 2,000 marine species. So this is very interesting, and based on this data, the area is now being called a coral triangle. So it encompasses the area of Malaysia, the ocean of Malaysia, Philippines, Indonesia, so these 3 areas. So also including Vietnam and neighboring countries. So yes, I think this is why since the ancient time, this region is very important, both economically and strategic waterway for trading, as well as for producing goods for the world. So yes, there's a lot of activities, there a lot of resources that is important to be discovered. Okay, so when we focus on doing this drug discovery, we focus on the mangroves, the beds of seagrass. We also focus on the coral reefs, living organisms, as well as sediment bottom. So we have a team to do the sampling monthly and annually to collect samples from various places within this within our ocean territory. So in IMB, in Institute Marine Biotechnology, we have our own scuba diving team that is doing the diving work. Yeah, they are really an expert. Okay, so apart from the coastal line and the shoreline where we collect our samples, we have one particular island, which is Bidong Island Research Station, belongs to University Malaysia Terengganu, where we do a lot of sampling work. Underwater sampling work. Interestingly, this island has been gazetted for no fishing, no diving, no tourism activities, so that this island and its surrounding area is well preserved, mainly for ocean studies. If you come to University Malaysia Terengganu, I'm happy to bring you to our Bidong research stations. So you can do sort of snorkeling and diving over here. Okay, so sampling strategies. This is what we are doing every year. Okay, so if the sample is in the deeper ocean, so we have to use our vessel. So these are our activities. If we looking for coastal plants, we go to the forest nearby the ocean to collect samples, and of course we do a lot of underwater sampling by our divers, and also we collect samples in the deeper ocean using our research vessels. We call it RV Discovery. So we are the only university in Malaysia to have this research vessel. Okay, so these are part of the activities that we are doing. We also do some studies regarding corrosion, regarding surface colonization underwater. And it is we are able to do this because we have the team that can dive in the water at any time. So unlike in the on the terrestrial land, of course the research is a bit expensive though. Okay, so there are few exploration works that we are currently doing in our institute. Number one is microbial exploration. We have a lot of microbes that living together with the sponges and other marine organisms underneath the water, as well as along the mangroves plant. So we isolate these endophytes microbes, and we isolate and to see, classify them, and look into the potential metabolites that being produced by the microbes that we collected. So another activities that we do is a chemical exploration. In this chemical exploration, we collect samples from marine organisms as well as mangroves and coastal plants, and then we do make a crude extraction using hexane and also solvent, other solvent, and then from this extract we further fractionate into active fractions, and from the fraction we then do the further analysis to look into the specific compound that is present in the fractions. Okay, so another exploration that we do is the DNA exploration. Most of the organism that we collected from the ocean, we isolate their DNA in order for us to make identification, as well as for further studies related to DNA work, including the potential of doing DNA insertion, and also do the all the molecular related work. Okay, so these are the example of the samples that we collect, and we fix and we keep in our repository. Okay, so the work is very lengthy, although it is like a routine work, but we follow strictly to this repository workflow, so that it is the workflow is important, because we want to have a very systematic method for the discovery work. Okay, and these are the example of the sample ID that we collect. We use for the identification of the samples that we have in our repository, and this is also the sample storage coding that we have. Yeah, so if you have a chance to come to our university, we are happy to bring you to our facilities. Okay, so as I mentioned before, the crude samples, the fractionation, the active fraction, or what we call the fractions that we separated from the crude extract of the samples, we then do the screening for cytotoxic effect using HeLa cells and HepG2 cells. So any fractions that give the value of IC50 below 30 microgram per mil will be selected for further studies. And for this, we are using MPS bioassay, and this method is used throughout our screening process. And the laboratory, the animal cell culture laboratory that where we do the cytotoxicity study is actually accredited under ISO 17025, and this is a valid certified toxicity testing. So okay, as for now, in IMB we already tested over 1,300 fractions of samples, including 350 fractions from coastal plants, 50 fractions from corals, 90 fractions from echinoderms, 240 fractions from mangroves, 170 fractions from sponges, 30 fractions from seaweed, and 350 from terrestrial plants. So among 1,300 fractions, we already identified 34 fractions to be highly potential for further studies. Okay, so those are from this species. Yeah, I'm not going to name everyone, but we have Aptos, and we have also Stichopus. This one and Holoturian is actually we call it sea cucumber species. Yeah. Okay, moving forward, these are actually the IMB marine biota. This is where we keep our samples and our fractions. Okay, and then we also established the data management site, which is www.neptics.net. In this Neptic, we provide all the information regarding the samples that we collect from the ocean. And this is the national, this is a Malaysian referral database for marine sampling activities. Sorry. So, okay. From that 34 important potential fractions that we have, we then further isolate and then discovered a few interesting compounds. For example, we have aptamine, 9-methyl aptamine, and so on. So this was discovered in the fractions of Aptos, and then it has been discovered to have potential as anti-cancer agent and also to reduce the progression of atherosclerosis. So, and also we found that another samples to have anti-cancer activities. Okay, so of course there's a lot more to be done in order for us to find and to develop drugs for marine resources. And I would like to share, we are very active in doing discovery works, especially on the structure functions studies. So these are some of the publication that has been published by our team in Institute Marine Biotechnology. For example, This is the work of Associate Professor Dr. Habsah Muhammad. She's looking into the structure functions of marine compound for PCSK9 inhibitory activity, and also anti-HSV activities, and so on. So also I just took some work from Dr. Yossi as well. I think after this, in the invited speaker session, Dr. Yossi will talk more in detail about her research work regarding the coastal plant research for drug discovery. So these are some of the works that I can share with you. Of course, there will be a lot more, because every year we are publishing around 100 papers, more than 100 papers a year in the internet, in the Scopus and WOS journals. Yeah. Okay. So I would like to end my presentation by concluding that there are enormous opportunities in marine discovery of the tropical ocean, as what we are doing now, and the effort to find a novel drug from the marine resources requires structured multidisciplinary sciences and technologies. And what we found also, that the institutional research collaboration will indeed speed up the journey towards marine-based drug development. So with that, thank you very much for your kind attention. Over to you, Madam Moderator.
Thank you, Professor. Last but not least, our fifth keynote speaker would be Associate Professor Dr. Arif Gusnanto from Department of Statistics, University of Leeds, United Kingdom, with his presentation title is Statistical Methods on Genomic Copy Number Alterations. Before we proceed with his presentation, let me give a brief summary of his current research. Dr. Arif Gusnanto is a statistician whose research focuses on development of statistical and data science methodology and inference in many areas, including bioinformatics and those with high dimensional data. He is the Deputy Project Coordinator of IM4 Future, a European Union funded training network with a budget of 2.9 million euro, with 11 partners from 5 countries, with focus on development of statistical methodology and analysis on genomic markers and biomarkers for aging. He is involved in many research collaborations and supervised 16 PhD students on many areas of statistical methods and applications in data science and analytics. He is a member of Royal Statistical Society. His education in 2017 until present would be is Associate Professor of Department of Statistics, University of Leeds. And in 2000 until 2001 is PhD students in Statistics, Department of Statistics, University College Cork, Ireland. His relevant publication recently would be focused on properties and approximate p-value calculation of the Kramer test, and also the prediction of tumor pathological subtype from genomic profile using sparse logistic regression with random effects. So, with no further ado, we let Dr. Arif Gusnanto to present his paper. Thank you.
Thank you very much. How much time do I have? Sorry, can you?
20 minutes, Doctor.
20 minutes. Okay, let me. Yeah. So thank you very much for the invitation and the opportunity to share with you some of the research that we have done. This is the research that we have done in Leeds, in particular in the development of statistical methods for omics data. So allow me to share. All right, one moment.
Oh, sorry, doctor. Once again,
Yeah.
what do you ask?
So can you see my slides? Yeah.
Yes, of course. Yes.
Okay.
Yes.
Right. So thank you again for the opportunity. So I'm going to talk about statistical methods on genomic copy number alterations. And I think there's a PhD student from Leeds who will give a talk in the invited session later on in the room mathematics with regard to this topic. So the purpose and outline of the talk is basically first to highlight the applications of statistical methods in dealing with a problem of biomedical science, which is basically copy number alteration and cancer. So today I will talk about detection of copy number alteration and prediction of tumor subtypes. And if time permits, I only have 20 minutes or less, we talk about prediction of cancer patient survival. So the motivation is that, first of all, the lung cancer develops over many years and stages. Okay? So they don't just become a cancer. They start from lesions and things like that, and it's developed into cancer. And usually there is an alteration or changes to the chromosomal copy numbers that drive the invasion. Okay, so as you can see here in the left panel figure, so this is basically chromosomes in normal cells. Yeah, so we have chromosome 1, 2, 3 up to the sex chromosome, and as you can see, each of this chromosome have 2 copies. Okay, so that's on the left figure. On the right panel, as you can see, this is chromosomes in lung tumor cells. And as you can see here, that in chromosome 1, for example, instead of 2 copies, it has 3 copies. And not only that, the chromosome 1 is actually shortened in a way. So some sort of deletion or a gene loss take place here in lung tumor cells. So this is what we are going to characterize. So as you can see here in the top, if we have 2 copies of chromosome copy numbers, they will appear as 2 copies for the whole genome. If you see the middle row here figure, if there is a part of the chromosome that is deleted in a way, meaning that at the beginning of this chromosome you only have 1 copy, and then the remaining of the chromosomes have 2 copies, it will appear like that as well in the chart on the right column. And if there is an amplification somewhere in the chromosome, it will appear that it has more copy numbers. So if you can imagine, if I divide the number of the copy numbers of the middle and the bottom figures here with regard to the normal copy number, you can see here on the right panel, we have ratio of half and then ratio of 1. Yeah, so an increase of ratio, a drop in the ratio at the beginning of the chromosome, and then a ratio of 1 for the rest of the chromosome. The same goes for the bottom panel here. It goes as ratio 1, meaning normal copy, normal 2 copies, and then the ratio 2 as 4 copies, etc., and then drop again. Why are we interested in this? Well, basically we are interested in the location of cancer genes, whether it's oncogenes, meaning genes that proliferate cancer, or loss, which is basically tumor suppressor genes. And there are different methods to obtain or to measure or to estimate copy number. But what we are interested in is something called next-gen sequencing or high throughput next-gen sequencing. So basically what happened is that if you have a DNA, yeah, so we chop the DNA in the sequencer in a way, yeah, we chop them. So these small fragments of the DNA called reads are mapped to human reference genome, and then we bin them, or we count how many number of reads in a fixed window in the genome. Yeah. So you can imagine that if we count the number of reads in the tumor and the number of reads in normal sample, yeah, we will have a ratio of half, and then 1, and 2, as you can see here. However, this is an ideal situation. In reality, that's not what we see. So here, this is a patient, yeah, LS041. On the left here, this is basically the copy number ratio for chromosome 1, and on the right is chromosome 8 from the same patient. Okay, so we don't see clearly what is the ratio, and in particular, because they have different number of read counts, we don't know to which copy number ratio should we assign each of these segments, as you can see here. And this is due to random error, different genome sizes, etc. And later in the talk by Maharani Umi, she will talk about how to use statistical methods to delineate the biological random error that you see in the figure like this. Yeah. So this is the data from patient 041. As you can see here on the top panel, these are basically copy number ratio, and it's difficult to assign to which copy number the segment should be, yeah, because in terms of ratio, it's not clear, yeah. But when we perform some sort of normalization, yeah, many statistical methods involved in this step, yeah. In the normalized scale, we can see here in the solid line which segment correspond to which copy number ratio, yeah. So if you see the bottom panel here in chromosome 1 only, on the left figure, bottom left figure, we see it's difficult to identify the segment, but on the right bottom right figure, we can see that each segment are mapped to the expected copy number ratio. Yeah. So we use this type of data for prediction of tumor subtype. Yeah. So there's a tumor stratification involved. In terms of personalized medicine, this is important in recent years. This is because precise histological substratification, meaning the right prediction of tumor subtype, is critical because the treatment that the clinician or the doctor give to the patient really depend on this histological substratification. Yeah. So we have 76 lung cancer patients in this study from Leeds Teaching Hospital, and comprising of two groups, basically squamous carcinoma and adenocarcinoma tumor. Yeah. So the aim here is, can we build classification models for prediction of lung cancer subtypes? Okay. So we have two types of data. Yeah, so these are basically the same patient. So if you see this, the top and bottom panel, this is basically of the same patient. It's just that they are estimated using different methods, yeah, different statistical methods in terms of copy number. The top one is using something called smooth CNA. So it manifests as a smooth line between genomic regions, as you can see in the top figure. In the bottom panel, this is what we call DNA copy CNA, where it uses segmentation using a particular method called circular binary segmentation. So it estimates copy number as fragmented segments. Yeah, so this is basically two different statistical methods to estimate the change points, yeah, in the genome. So what we do here is, for the classification, we consider the logistic regression. For those of you who come from statistics or mathematics background, allow me to explain what's going on here. So basically, the tumor subtype, whether it's coded as 0 or 1, is assumed to follow a binomial distribution with mean mu. So this mu through some link function is modeled as x times beta plus z times gamma. So the information, clinical information of the patients are basically contained in x with parameters beta, and the information on copy number alteration is basically contained in the matrix Z here with parameter gamma. So gamma here is assumed to follow a normal distribution with mean 0 and variance tau square. So you can imagine that because in copy number alteration, we have such a high dimensional data with 17,000 genomic regions. So the matrix Z here has approximately 17,000 columns or 17,000 variables. So gamma here, as the parameter of associated with Z, is a factor of 17,000 elements. Yeah. So basically, we use penalized regression. So the joint likelihood of the parameters here is basically penalized by square penalization. In the recent paper that we published, the penalization here is more complicated than this one. I haven't put it in this slides, but it's more complicated, which produce sparse solution, as the moderator suggested. But in this particular instance, this is not a sparse solution. It's basically a simple penalization, and the estimation of model parameters basically solve this mixed model equation here in the bottom of the slide. Yeah. This is the result of model fitting an estimate for the fixed effects and the clinical information in terms of age and gender without CNA profiles, with smooth and with the DNA copy CNA profiles. So as you can see here, that there is no significant differences between the probability of tumor subtypes in terms of age and gender, because the p-value here is all of them are above 0.05. So in terms of the estimate, gamma, as you can see in the previous slide, so the estimate of gamma is plotted in this figure. In the top panel, this is using smooth CNA data. Yeah. So we can see here that all of them have positive, negative estimate that indicates, if it is a positive estimate, that a high copy number is associated with the probability of the tumor to be squamous carcinoma, yeah, and a negative estimate for each of the genomic region here. A negative estimate correspond to higher probability to be adenocarcinoma, yeah. The same goes on the bottom panel for DNA copy data. One thing to note here is that in chromosome 3, there is a region in the second half of the chromosome here that shows a distinctive pattern that a positive value in that region correspond to probability, increasing the probability to be a squamous carcinoma. The model is able to separate, in a way, the patients in those with squamous and those with adenocarcinoma. So for those of you who are not familiar with this type of plot, on the left panel here is basically based on smooth CNA data. The horizontal axis is the probability of the tumor to be squamous, and in the vertical axis, this is the observed value, whether it's squamous or adenocarcinoma. So as you can see here, they are quite distinct in terms of grouping. Yeah, the same goes for the data based on DNA copy CNA. Yeah. In terms of cross-validation, meaning how well the model will be able to make prediction on copy number alteration, the proposed method is basically here, the left one, denoted as log, as in logistic model. It has a quite low classification error. The median value is around 2-point-something% there. Yeah, it's less than 2.5. Yeah, around 2%. Sorry, around 4% actually, because this is classification error in terms of number rather than percentage. My apologies. But if we see the other classification methods, yeah, basically from the left to right here, we have K nearest neighbor with one neighbor, K nearest neighbor with two neighbor, discriminant analysis 1 and discriminant analysis 2, linear and quadratic, PLS, neural network, lasso. Sorry, this is elastic net, not neural network. Elastic net, and then lasso, and then neural network, support vector machine, and then smoothed logistic regression. So it has a quite low classification error, and in fact, this method is actually being used in the hospital to help pathologists in making a decision. Yeah. So that's basically the result that has a quite low classification error. Due to the time, this is already 19 minutes. I have 1 more minute left. I'm more than happy to discuss about survival analysis in this case, but allow me to jump to some concluding remarks. Statistical methods are needed to make sense of data as information, and in this context, this big data or high dimensional data that turn data into analysis, into result, into insight, and into action. But this is not unique for biomedical areas. We in Leeds are involved in many applications of statistical methods in biology, medicine, engineering, etc. And it's basically have modern experiments with complex data. So I think that's pretty much it. So this is already 20 minutes now. We thank the surgical teams at St. James Hospital Leeds and Yorkshire Cancer Research who supported this research. Some members of the department that also involved in this research. So thank you very much for your attention. I'll be giving this back to the moderator. Thank you.
Thank you, Dr. Arif Gusnanto. Okay, this is the end of presentation from all keynote speakers. For the next session, would be question and answer for from participants to all keynote speakers here. First of all, for all respected keynote speakers, please provide us your presentation slides by sending to chairman emails as soon as possible. Thank you. So just reminding for provide us the presentation title. Presentation slides, what I mean. Okay, for next would be the question and answer session. We already collect all the questions from the chat box. And also if participants feel that she or he would like to ask directly to the speakers, then can raise hand to be chosen by the operator. Thank you. So meanwhile, I would like to read some questions. First, for Professor Marcelo, the question from Adi Rahmat, University of Sriwijaya. I have a question regarding your material. Is it working as gas sensor specifically for nitrogen dioxide? Can your material senses other gas such as sulfur dioxide? During dry season, we occasionally face land and forest fire. This fire releases a massive amount of smoke. Is there any sensing material suited for this? The smoke can very thick. Okay, Professor, I think you can just give brief explanation about this answer. Thank you.
Yes, thank you very much for the question. And yes, for the fire, we have the sensing for CO. So it is byproduct of fire usually when you have it. And about the sulfur, we are not working on it because we have a problem. It is necessary a dedicated system, because if you try to measure sulfur and after is going to use another analyte, you have a stronger contamination. So we are not using any kind of sulfur on our gas measurements. But about CO, yes, we can detect also CO. I didn't show the results, but we have materials to detect CO.
Okay, we move to Pak Adi Rahmat. Is it any further question, or do you satisfy with the explanation from Professor Marcelo? Okay, we move on to next question from Handoko from Institut Teknologi Bandung. Requested question to Professor Puji. How about the absorber ability to remove dissolved solid, such as coloring agent, released by textile industries?
Okay, thank you for your the question, Mr. Handoko. Nanomagnetic and nanomagnetic modification can be used to absorb dye from the textile industry by several mechanisms. Physical interaction if the nanomagnetic coating with porous material, for example, activated carbon, bentonite, zeolite, and the other mechanism if the nanomagnetic coating of the material interaction by electrostatically. Dyes in solution are anionic or cationic dyes. Then in the solution can have a positive or negative strength. The nanomagnetic material have a pH PZC 0.0 change. If the dye is anionic, then the electrostatic interaction is more effective at above of pH PSJ. Or if the dye is cationic, interaction effective above pH PZC. I have used nanomagnetic to absorb wastewater from the songket industry. Songket is traditional clothing by the people of South Sumatra, Indonesia. In the coloring process, many synthetic dye are used. The result is quite effective in reduction dyes. Okay, thank you, moderator.
Okay, Professor Puji, the second question from Desnelli, Sriwijaya University. In the slides described, various methods are used for nanomagnetic synthesis and nanomagnetic modification. What is the best method to use and what are the criteria for a good nanomagnetic result?
Thank you, moderator. I answer to the question. There are many methods for nanomagnetic synthesis, such as co-precipitation, sol gel, hydrothermal, sulfothermal solution combustion. Each method has an advantage and disadvantage. Each method also has a variable that determines success. For example, as using a co-precipitation method, the success of nanomagnetic synthesis is high, but the time is long. The product depend of the precursor use for and concentration of sodium hydroxide, temperature, and speed of addition of sodium hydroxide. If the addition of sodium hydroxide must be little by little to produce a small crystal size so that the magnetic properties are like with the surface area is less. As use the combustion solution, nanomagnetic synthesis needs fast time. The success of synthesis depends the fuel as used. Because the method of nanomagnetic synthesis or modification of nanomagnetics depending on the cost, equipment, and product we expect. We hope that the nanomagnetic and nanomagnetic modification product are expected to have small crystal size, large surface area, and large magnetic properties. Thank you, moderator.
Okay. For the next question, again, Professor Puji, we have our third question from one Rian Asri from Sriwijaya University. Also, he mentioned here, as we know that titanium dioxide is a heterogeneous catalyst for that is often used. So in this context, titanium dioxide is used as a double coating for nanomagnetic. So these questions, first, is there any significant difference in the photo degradation efficiency if titanium dioxide is used directly and titanium dioxide as a shell? And the second one would be based on the presentation, silicon dioxide is used as an intermediate coating. What if titanium dioxide is used to coat nanomagnetic ferrite without silicon dioxide? Thank you.
Thank you, moderator. Titanium oxide is a heterogeneous catalyst that is often used. The use of titanium oxide as a catalyst has drawbacks. Number one, titanium oxide to form a suspension in solution, so that is difficult to separate it after the degradation, photocatalytic degradation process. And number two, titanium oxide can only be used in the ultraviolet region because it has a large band gap. Titanium oxide have band gap around 3.5 electron volt, not only in the UV ultraviolet, but also visible region. And the second question, answer for second question. Silica intermediate is passive layer can effectively prevent degradation and dissolution of the magnetic core and promote the photocatalytic activity by reducing the adverse influence of the magnetic core on the photocatalysis of titanium oxide. Direct contact between ferrite compound and titanium oxide causes titanium oxide to enter the core and then oxidizing the iron. And consequently, the ferrous iron or iron ion dissolve into solution. The advantage of this modification are a magnetic core for separation in magnetic field and interlayer preventing photodissolution and carrier recombination. Thank you, moderator.
Okay, thank you, Professor. Next to next presenter, the question from Bapak Hermansa from Sriwijaya University to Associate Professor Walid Fuad. What kind of method can reduce lignin content effectively?
Thank you for the question. Lignin, as I mentioned briefly in the chat, lignin is very essential for the plant. So it's very important not to demolish lignin completely, because lignin is involved in plant tolerance to disease and resistance to pathogens, things like that. So the best methods will be kind of targeting by synthetic pathway of the lignin, and there are like multiple. It's quite complex pathway. So certain genes, mutation in this certain genes, reducing the expression of the gene or mutating the efficiency of the enzyme would be sufficient enough to reduce the lignin, but not demolish the lignin, and to the level that actually will improve the hydrolysis of the cellulose fibers. So RNAi site-directed mutagenesis, both of them can work efficiently.
Okay, for the next question from Eliza, Sriwijaya University, addressed to Professor Fazila. As we know that Indonesia has marine too, but our problem is to determine the marine. Do your university has herbarium to determine the species of the marine? Yes.
Okay, thank you for the question. Of course, in our university, we have herbarium for marine samples, and we also have a few experts who can identify marine species.
Okay, again, Professor, for the second question, have you produced products that have been marketed from mangrove research?
The answer is no. There is not yet any medicinal-based product from mangrove, but we have studies quite intensively, especially in the terms of extract that is high in antioxidant properties.
Okay, Professor. Next question from Muharni, Sriwijaya University. Again, question to Professor Fadzila. Have you ever found antioxidant compounds that are not phenolic or flavonoid groups? If so, what are the compounds?
Okay, to answer that, because I didn't do antioxidant studies, but our group has done a lot of antioxidant screening for most of the fractions that we have in our collection. But I have to, as I already wrote in the chat box, I'm happy to ask our team in IMB and let the question be answered as soon as possible. I will have to refer to our team.
Okay, thank you for the information, Professor. Last question so far. From Dina Akfar Yarsi from Pontianak. What are the challenges in maintaining marine health sustainability in marine research?
Okay, if regards to the way we handle the samples and the way we collect the samples at the site, of course, we adhere a very important regulation, strict rules, whereby first there will be a minimum number of divers that can go for sampling, because we don't want to disturb the environment. The second one is that we only select samples that are abundant, that grow well in the site that we are doing the sampling. And of course, when we collect the samples, we only specifically collect what we want. We don't simply take any samples just by looking at it, it is beautiful, then we take. We already predetermine the samples that we want prior to going for diving. And also the sites for our sampling area are the sites where it is a restricted area. Usually in our country, there are a few restricted areas whereby there will be no fishing, no diving activities, what we call it, recreational tourism activities being allowed at certain area for our that has been allocated for research. So that is how we try to maintain our marine health environment. But of course, there are challenges. For example, when we do the diving at in one site, whereby when we come next time to collect more samples, there will be none, because underwater, tidal, and there is a lot of movement in underneath the water, underneath in the seabed, so that we also find quite challenging, whereby we lost the sample, then we have to select a new one. But usually we rely on the GPS system that we already to marking our sampling site. Okay, those are some of the answers that I can share.
Yeah. I hope all the explanation from Professor Fadzila could satisfy your eagerness to know a little bit about the topics. So for next question, again for Professor Puji Lukitawati from Widya Twini Rizki, Sriwijaya University. From the explanation, we know that nanomagnetism and nanomagnetic modification can be used to inhibit bacterial growth. So the question here first, can you explain about the mechanism of inhibiting bacterial growth? And also the second one is the nanomagnetic only used for inhibit bacteria in the environment, or it can be used for biomedical application?
Thanks for the first question. I have two questions. And for question one, nanomagnetics exhibit antibacterial properties due the reaction with the surface of the cell wall. Irradiation of photon with energy to the nanomagnetic or modification of nanomagnetic causes electron excitation from the valence band to the conduction band. And furthermore, the hydroxyl, perhydroxyl, superoxide, anion radical can be procreated by interaction between the dissolved oxygen and electron that increase inhibitors of bacterial growth. The attack of hydroxyl radical to the bacterial cell wall actually lead to the breakage of the structure. Another mechanism that causes nanoparticle antibacterial activity is this change of reactive oxygen species, such as superoxide and hydrogen peroxide, from the surface of nanomagnetic or modification. This active oxides amount have an efficient role in bacterial eliminating by nanoparticle through the penetration in their cell wall. And then the interaction of nanoparticle which have a positive charge with the bacterial cell that has a negative charge leads to leakage of the bacterial cell contents. Ferrite compound nanoparticle indicated substantial antibacterial activity of this metal ferrite biocompatibility and appropriate option for antibacterial use in industrial and medical fields. The particle, for example, iron oxides, already understood to be non-hazardous magnetic material, and also I contrast again, have already proved as harmless for human use. Photocatalyst is used for various fats such as cancer cell and bacterial infection is not harmless. Thank you.
Okay, for the last question, so sorry, we almost run out our time, so we have to move to other session. So for the last question, for Dr. Arif Gusnanto, there are two questions here. First one from Dian Cahyawati, Mathematics, Sriwijaya University. What the criteria models that we have to explain in using logistic regression, and are there any assumptions for logistic regression? And also the second one from Professor Fadzila, can you share a little bit about your research related to aging, since my undergraduate biotechnology is also from Leeds University. Thank you. Please, Dr. Arif.
Right, thank you very much for the question. With regard to the logistic regression, the use of logistic regression here is due to the assumption that the label of tumor subtype coded as 0 and 1 to follow a binomial distribution. So the construction of the whole model used kind of like a similar principle as in generalized linear model. The assumption that we consider important in this particular modeling is actually the assumption on the parameters gamma, as we see earlier. Because our assumption that gamma follows a normal distribution, we arrive at a solution or parameter estimates, as you see in my slides, which is kind of like estimate for each genomic region. Now, in our more recent paper, when we use a different assumption on the random effects, we are able to produce sparse estimate, meaning that estimate that only shows non-zero estimate only on some of the genomic region. Okay, so this is something that we call sparse modeling, sparse statistical modeling, which I'm more happy to discuss further if you wish. With regard to the second question on the research related to aging, I'm sure some of you may have might be familiar with. We have two different age actually as a human being. First is the chronological age. So this is the age that we usually count from our identity card. Yeah. So we know when you were born, and you just calculate the time between when you were born and today to know your chronological age. But there's something different in terms of age, which is what we call biological age. With regard to biological age, there is a marker called N-glycomics, yeah, in our body. So it is possible that our biological age is actually less than our chronological age. Yeah, so you are lucky. That's why we see some of the participants very youthful. Okay, so you look like you're still a teenager, even though you are 30 years old probably now. But some of us also have biological age more than our chronological age. Yeah. So probably me with a lot of gray hair at the moment looks older, much older than my my chronological age. This is just an example where probably biological age is more than chronological age. So our research now works on how this glycomics data are used to determine many aspects in our biological age, including why some patients experience Down syndrome, for example. Okay, Down syndrome is an example of accelerated aging. Okay, so this is just basically some of the research that we do. Of course, it's going to take a lot of time to explain this in details, but basically, in principle, that's what we do in our current research. So thank you, the moderator. Yeah.
Okay,
Thank you very much.
thank you. Okay, thank you again for Professor Marcelo Ornagi Orlandi, Professor Dr. Puji Lukitowati Haryani, Associate Professor Dr. Walid Fuad, Professor Fadzila Adibah binti Abdul Majid, and also Associate Professor Arif Gusnanto for answering those questions and for the great presentation. It was a pleasure to have you with us. So this concludes the presentation. Thank you for all the attending. We hope you have learned and enjoy this presentation. So I will give it back to the Master of Ceremony. Thank you. Sorry for the all respected keynote speaker. Once again, don't forget to send the presentation slide to the chairman email as soon as possible. Thank you.
Sorry, can you share the email address in the chat, please, so that we know to which email address to send? Thank you.
Okay. So last but not least, sorry, forget. Give participant, give applause, big applause to all our respected keynote speakers today. Thank you.
The next agenda is break time. To all audiences, please take a break.
Sriwijaya University. Sriwijaya is one of the state universities in South Sumatra, with 2 campuses in Palembang City and Indralaya Ogan Ilir Regency. This university comprises of 10 faculties and 1 postgraduate program. One of the faculties is the Faculty of Mathematics and Natural Sciences, commonly known as a Faculty of MIPA. Faculty of MIPA of Sriwijaya University was founded on March 6, 1989 with the name Mathematics and Natural Sciences program under the Faculty of Engineering. By October 22, 1993, the program had 4 study programs, namely mathematics, physics, chemistry, and biology. During the 1993, the Mathematics and Natural Sciences program changed to the Faculty of Mathematics and Natural Sciences on October 22nd, 1993. For academic year. Namely the Marine Science Study Program, which was established based on an operating permit decree from the Directorate General of Higher Education. Then in 2011, the Pharmacy Study Program was added. In 2014, the faculty established the Physics Master Program and the Mathematics and Natural Sciences Doctoral Program. Finally, in 2017, the Postgraduate Program of the Faculty of Mathematics and Natural Sciences added 2 study programs, namely a Master of Chemistry and a Master of Biological Sciences. Therefore, currently, the Faculty of Mathematics and Natural Sciences has 6 study programs at the undergraduate level and 4 study programs at the master's and doctoral level. The study programs at the undergraduate levels are as follows: Mathematics Study Program with Accreditation B, Physics Study Program with Accreditation B, Chemistry Study Program with Accreditation A, Biology Study Program with Accreditation B, Marine Science Study Program with Accreditation A, Pharmacy Study Program with Accreditation B. And then, at the master's level are as follows: Master of Physics has Accreditation B, Master of Biology has Accreditation B, Master of Chemistry has Accreditation C, Mathematics and Natural Sciences Doctoral Study has Accreditation B. The expectations of the Faculty of Mathematics and Natural Sciences of Sriwijaya University are as follows: the creation of graduates able to master and develop knowledge in mathematics and natural sciences, the creation of graduates with the ability to compete and meet labor needs in society, government, and industry, producing research by lecturers or students in the form of empirical knowledge, theories, concept, methodology sciences. The realization of the Faculty of Mathematics and Natural Sciences of Sriwijaya University as a Center for Services and Information, Deputy Dean for Student and Alumni Affairs. The Faculty of Mathematics and Natural Sciences of Sriwijaya University has students, organization, and autonomous body study program such as Faculty of MIPA Student Executive Boards, Student Family Representative Council, Student Association for Environmental and Adventure Activity Mapala Sandi Rimba Kami, Muslim Scientist Generation Community, Community of Science, Sriwijaya Diving Club, Himastic or Mathematics Student Associations, Himafia or Physics Student Association, Himaki or Chemical Student Association, HMB or Biology Student Association, HKMF or Pharmacy Student Family Association, Himaikal or Student Association of Marine Science. Faculty of MIPA has various facilities for all departments. such as the department has a large and luxurious hall courtyard for all student and faculty activities, Hiyaki bus and cars, multimedia room, conference room, student organization room, ICT room, business and innovation incubator room, entrepreneurship center. Furthermore, there is also an Musola facility for all academicians. It also has sport facility, namely futsal and basketball courts. It also has a butterfly house, and it also has a classroom are comfortable and cool. Each department has a reading room and laboratory with a accredited laboratory comprising and sophisticated equipment such as AAS, FTIR, echo sounder, CTD, spectrophotometer, HPLC, and etc. Each study program has a lecturer, seminar, and meeting rooms with professional educators, including 7 professors. The vision of the Faculty of MIPA of Sriwijaya University is to make the Faculty of Mathematics, Natural Science a faculty excelling in science and technology by 2025. The Faculty of Mathematics and Natural Sciences of Sriwijaya University has the following mission: to organize education in the field of science and technology, to produce graduates are able to apply science and technology to the welfare of the nation, to organize research activities in the field of science and technology, to organize community service activities in the field of science and technology, to apply science and technology that is beneficial to society, to establish strategic cooperation relationship with external parties at the national, regional, and international levels. The faculty's motto is grow, develop, excel, and prosper together.
Ladies and gentlemen, let's resume to the previous agenda. The chair of this session is Mr. Gusti Diansyah, Master of Science. To Mr. Gusti Diansyah, time and place are yours.
Oh, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Here we are in the second session of this conference. The invited speakers' presentations in the second Sriwijaya International Conference on Basic and Applied Sciences. I'm Gusti Diansyah from Marine Science Department of Sriwijaya University. First of all, I would like to greet our Rector of Sriwijaya University, Professor Anis Sagaf, MSc, and our Honorable Dean, Faculty of Mathematics and Natural Sciences, Mr. Hermansyah, PhD, and of course, the Honorable all of invited speakers. There are 5 speakers who already joined with us today. First, Dr. Dede Heri Yulianto from Research Center for Biomaterials, the National Research and Innovation Agency. And the second speaker is Mr. Acep Furkon, PhD, from Institute of Technology Bandung. And the third speaker is Ms. Dr. Yosi Handriani from Institute of Marine Biotechnology, University of Malaysia, Terengganu. And the fourth speaker is Associate Professor Dr. Zailan bin Siri from Malaya University. And the last but not least is Ms. Endang Linirin Widyastuti, PhD from Lampung University. And of course, to all of participants who already joined with us, we are very pleased to see you here and welcome to all of you to this spiritual conference. Ladies and gentlemen, before this presentation begins, let me inform you how this presentation will be going on. First, all speakers will be invited to present their presentation, and every speaker will have 15 minutes to deliver their speech. And after all speakers already delivered their speech, we will have next session. It will be discussion, question and answer, and followed by closing session. Ladies and gentlemen, right now, allow me to invite the first speaker, Mr. Dr. Dede Heri Yulianto. Good morning, sir.
Yeah, good morning. Thank you very much for invitation.
Yeah. Before Dr. Dede Heri Yulianto delivering his speech, I would like to read to all of you his curriculum vitae briefly. Mr. Dr. Dede Heri Yulianto has been work in Research Center for Biomaterials, Indonesian Institute of Sciences. I mean, now it's already changed, I think, to the National Research and Innovation Agency from 2015. He did his bachelor degree in IPB University in Analytical and Environmental Chemistry, and he did his master degree and PhD degree in Ehime University, Japan, and especially in Applied Bioscience Department. Joined with him is already more than 26 in total to date selected peer-reviewed publication, and also it's quite huge number and patents. It's quite amazing, Pak Dede. It's more than I think it's almost 13 patents, 2 Japanese patents, 1 WIPO patents, and 10 Indonesian patents. It's amazing. And now, Dr. Dede Heri Yulianto, time is yours.
Okay, thank you very much, Mr. Chairman. And I would like to thanks to the committee for this opportunity to share our research progress within these 15 minutes. I take the title Recent Achievement in Textile and Batik Industry Wastewater Treatments. And you know that almost 85 rivers in Indonesia is heavily contaminated with textile dyes, and you can see there in this slide that so many wastewater are directly discharged into the rivers. So it is estimated around 200,000 tons of dye stuff is expelled into the global environment every year. And as we know that they use some dye chemicals, sometimes anthraquinone or azo, diazo, naphtha, and also indigosol dyes that originally designed to be recalcitrant for long-term use. So if this still occur, it is real dangerous for the environment because the azo dyes, for example, is resistant to aerobic wastewater treatment. And the textile dye wastewater are toxic and mutagenic and also carcinogenic. More than 100 dye compounds are reported as a carcinogenic chemicals such as azo and nitrous compound. And some techniques are already researched by the researcher, such as the physical methods or chemical methods and biological methods. In this presentation, I would like to review some methods and what methods will be very convenient or efficient methods for this textile dye wastewater treatment. The physical methods usually using the absorption. I think Professor Puji already presented his discover technology to treat the dye wastewater. They in this physical methods like absorption, the dye will absorb into the materials, and the other are using the membrane filtration, and the water will release into from the membrane, and the dyes will stay in the membrane, and we will get the clean water. Another technology is using the flocculant and coagulant technology by using the chemicals, and the principle of this technique is how the dye are trapping and then precipitate in the bottom or in the bottom layer. So another technique is using chemical methods such as the advanced oxidation. The Cato et al. 2080 use the zero valent aluminum to oxidize textile dust wastewater, resulting to the removal almost 94.4% and color removal until 80%. Another technique also using the ozonization by using the ozone, then the pollutant will oxidize into the oxidation product, or also sometimes using the Penton reaction. This is all the chemical methodology. So my research field is about the biological methods, how the biological methodology can also contribute to the removal of textile dyes. So the textile wastewater can be degraded. In this case, not only absorbed or oxidized by the chemical or by the physical method, but in this method, the textile dye will biodegraded by the microbe, for example, the fungi or bacteria, and also by using the enzyme directly to non-toxic compound. So according to previous, I described that the limitation of different techniques types, such as the chemical methods, has an advantage like a simple and also rapid and efficient process and multiple approach, but it is sometimes difficult to use in for the small and medium enterprise for the economically, because it is sometimes high cost, and we use a chemical also to add in the water environment. By using the membrane filtration, it is a rapid method and also straightforward, suitable for almost type of dyes, but it is a high energy and maintenance. You will need to clean every time the membrane because of clog resulting during the process of the filtration. By using the coagulation or flocculation, also this is a very simple physicochemical process and very rapid process. But the disadvantage of this technique is will generate the slots and also further increase the process costs. So I have a very simple video about the flocculant and the coagulant technique that very rapid decolorization. In the center is in the jeans washing factory. Then we develop flocculant very fast decolorization for these dyes by using only small flocculant, and only in 1 minute we can directly remove the color. But as you can see that after the decolorization, the flock will remain in the bottom layer or sometimes in the top layer. This is the advantage of the flocculation method, giving the fast or the rapid decolorization and technique, but also generate the flow that further increase the cost to remove this flow. So yeah, this is the technology for the flocculation, and you can get a very simple and rapid decolorization and also non-toxic product. But once again, this technique is a very high cost because we need to synthesize the flocculation material that result in this high efficiency for the dye removal, and also the cost for the remove the flock. So what I study that the best technique is a combination between this absorption and also the biological method. The absorption method is easy and simple and flexible operative and also economically feasible method for wastewater treatment. We can find various absorbent like low-cost absorbent based on the natural and agricultural waste and industrial waste like zeolite, chitin, clay, or also lignocellulose materials. And also we can also develop biosorbent or no carbon nanosorbent like a carbon nanomaterials like a nanocarbon, nanozeolite, and nano FaO4, FeTO4, and also halloysite nanotube. By using the biosorbent, we can develop a derivative from the biological source, like a microorganism. This technique, we need to analyze the absorption capacity and also maximum removal efficiency. And yeah, it can be as a cost-effective methodology for this absorption. And another technique is a microbial degradation. This method is a simple, economically attractive technique, can degrade the toxic compound to non-toxic compound, but the disadvantage of this technique is a slow process. Within 6 until 30 days, we can degrade this compound. So for this, we need research to find super microbes and how to immobilize this microbial into the absorbents with the combination. The other is we using the enzymatic biodegradation. It also can degrade toxic compound and to non-toxic compound. The process is fast in hours, but the challenge is how to reduce the cost production to produce this lignin, this enzyme. We can solve this problem by finding the novel enzyme or using the recombinant or also using the immobilization of this enzyme into the absorbent. Therefore, we propose that the combination of biological and physical methods is a efficient method to use for the wastewater treatment. So we combine the biological method and physical method by we isolate, screening, and identification of the potential fungi. We already isolate from the several places in Indonesia, and we select the best potential fungi to degrade the dyes by using the agar media or in liquid medium. And we then optimize the enzymatic degradation by using only the enzyme, and we immobilize the enzymatic and also the enzyme and also the mechalia into the biomaterial, different biomaterials, and we did the pilot application and implementation into the batik and textile wastewater. I think in this step, this number 4 is the key technology to how to produce the fourth-chain biomaterial. I mean the combination between the biology and the physical methodology to produce the green sustainable wastewater treatment. So why water fungi can degrade dyes? It is because the water fungi can degrade the lignin as one of the component in the wood, and the structure of the lignin is more complex if we compare with other dyes, chemicals, such as this is the reactive violet 5 dyes. So during this degradation, the fungi produce this manganese peroxidase enzyme, lignase peroxidase enzyme, and also lactase enzyme. In this case, I use the lactase enzyme how to produce by the fungi and use this lactase to degrade the dye wastewater. So we try to immobilize the best fungi into the material. In this case, I use the LECA, lightweight expanded clay aggregate. It is usually used as a plant media, and by very simple technique using the steam activation and washing, and we immobilize the fungi into the leca, and we call the product is mycoleca. So the mycoleca is contain the fungi inside and also outside, as you can see here. And we already apply into various synthetic dyes such as anthraquinone dyes, azodiazo, direct differs naphtha, and also indigosol dyes, and also various textile and batik wastewater from Kalongan, Purwokerto, Cilacap, and other place in Indonesia. And this is the color removal from the Purwokerto batik, indigosol batik. So the immobilization yield in this materials is around 68% and equal to 1.5 milligram of micellia per gram leca. So this microleca also can detoxify the dyes, and we have analyzed using the human dermal fibroblast cell. And from this graph, we can see that the treatment sample can non-toxic can produce the non-toxic wastewater after treated by the microleca. And also we compare using the phytotoxicity and also Artemia salina for the toxicity of these treated samples. Because of we using the biological approach, we can analyze the degradation of the dyes. And for example, this is the analysis of the anthraquinone dyes with the different brands of these chains, like using LB4 dye and also LBBR dye. Then we detect a very small compound, non-toxic compound during the degradation by the this microleca. So the microleca also have the excellent usability, and we already analyzed this for almost 20 times. And the degradation, the decolorization of this, the ability of this microleca is still around 100%. We added the new wastewater after decolorization. And also we detected the lacase activity produced by this microleca is also still occur during the decolorization, and we already conducted this in the Purwokerto batik and also in the Pekalongan printing batik for this test. And we also have applied directly into the wastewater into the batik industry in Pekalongan, and this wastewater is a very high COD, around 8,000 ppm, and based on this application, we can decrease the COD level around 1,000 ppm per day. And yeah, this is the result after 24 hours, and this is I got the report from the people there that the wastewater treatment is now almost successfully, because the fungi can by the acclimatization can degrade the wastewater very good after 3 weeks condition. And this type of the wastewater is a continue with a 3 until 5 type 5 cubic for days. And we also detected the enzymatic activity very easy in the field by using only for example, using the SBT. If the wastewater coloring the yellow, the green color, it means that the enzymatic activity is still occur and the fungi is still active to degrade the wastewater. We also have tried in one of the big company in Jakarta using this technology, and we obtained that the microleca can decrease the pH into the normal condition and COD, and also other component like a phosphate and also ammoniac. And also can absorb the total chrome during the degradation and the absorption by using the microleca, and reducing the toxicity until 83%. So we conclude that this microleca combination between the biological and physical method is a very efficient method for the wastewater treatment, especially for the textile dye wastewater treatment. I acknowledge my research group, Microbial-Based Technology, the National Research and Innovation Agency, BRIN. Thank you very much for your kind attention.
Oh, thank you very much, Mr. Dr. Dede Heri Yulianto, for such a remarkable presentation related to research on textile dye wastewater treatment techniques, including physical, chemical, and biological methods. And I think it's focused on in WRF, I think, White Road Fungi, how WRF can degrade dyes. Thank you very much, Dr. Dede, for presentation. Before we go to the second speaker, I would like to remind, it's a pleasure to, if the speaker would not mind to share to the community the presentation files. Thank you before. And now allow me to invite the second speaker, Mr. Acep Furkon, PhD. Good afternoon, sir.
Yeah, thank you very much.
Before Mr. Acep Furkon delivering his presentation, I would like to read his curriculum vitae briefly. It's from Bandung Institute of Technology in Department of Geophysics and Complex System, and also he also in Director of International Office in Institute Technology of Sumatra, and also Senior Collaborative Professor at Kanazawa University, Japan. And with him is already 55 publication at Scopus, and also he is the president of ForMind, Indonesian Young Researcher Forum. I think it's more than 200 researchers join with this forum, and also he is CEO of ForMind Institute. And now to Mr. Acep Furkon, PhD, the floor is yours.
Okay, thank you very much, the organizer. It's a great honor for us to deliver speech here. I'd like to share about our some current information about our research. Okay, we would like to talk about effect of temperature on surface deformation of two-dimension enzyme model using molecular dynamics of Langevin equation. As you know, in molecular dynamics, there are many methods depend on the point of interest. But some handicap about molecular dynamics, usually about the time, yeah, especially for all atom molecular dynamics. In biological function, it takes time. So we would like to discuss here to use other method using Langevin equation. Of course, there are some benefit, and then there are some handicap about this one. First of all, I'd like to thanks for organizer. It's a really good honor for us to join in the Sriwijaya International Conference on Basics and Applied Science. The Excellency of Rector of Universitas Sriwijaya, and also Dean of Faculty of Mathematics and Natural Science for this opportunity. So this is our outline: introduction, theory, method, and result, and discussion, and conclusion. So in our human activity, there are some activity about in drinking and eating, or work, and something like that. So many activity here, especially in our routine activity in food and drink. Yeah, there are some enzyme here, especially in early digestion in mouth. There are gland and enzyme. Yeah. And some food and drink condition, they are hot, cold, or something like that. Yeah. We would like to know about effect about in temperature, especially in enzyme activity. Yeah. Of course, there are many enzymes here. We just make a very simple model. That's only model. Yeah. Try to mimicking about some particular enzyme here, and we use Langevin equation, especially to know, yeah, surface of two-dimensional enzyme model. For example, here amylase enzyme, yeah, we try to make a model, just a model, yeah.
Excuse me, Mr.
Yes.
We couldn't find your file. Follow your presentation.
Oh, really? Okay, so sorry, maybe. How about this one? Is it okay?
Now working now.
Okay, sorry. So actually, we start from here, and then there are some our routine activity, especially during the digestion. Yeah. So we would like to know about the effect of the temperature, yeah, in here using Langevin equation. For example, here. So we try to make a simple model. For example, enzyme looks like yeah, they have their substrate and active site, and then we have a binding process, and we would like to know reaction result. Yeah. This is a simple illustration process in enzyme binding to a substrate. Of course, there are many methods we can investigate about this one, but for simple thing, yeah, we for educational purpose, yeah, we can make some simple one. As you know, this is a pioneer of Brownian theory of motion. Yeah. We, for example, Langevin approached the Brownian motion according to Newton equation of motion. As you know, there are complicated interaction, especially high cost in our time consuming. Yeah, if we calculate all interaction, of course we can get very detailed information about interaction, but in some cases we don't care about this one. For example, we just want to know the trend about it. So many interaction. Yeah, I mean with very wide frequency. I mean in their momentum they are very wide frequency. So the very smart idea from the Langevin try to make this as implicit interaction. Yeah. Using start from a Brownian motion. Yeah, Brownian motion, it's mean representing all interaction. I mean all interaction frequency. Yeah. On the basis stochastic properties, this is deterministic properties, and then change to Langevin equation. Yeah, and then we can see produce the theorem in fluctuation and dissipation. Dissipation is important part here. Yeah, and we can modify the Newtonian equation using Langevin equation. Yeah, there are some requirement to make it perfect. Yeah, as you know, for a long time there are some theorema here, and finally we can see the derivation until this one. And how about velocity? Yeah, actually we can derive the velocity part using this one. Yeah, and for t for infinite, we can find the simple expression for this one. And then for thermal equilibrium expressed by this one. And finally, we can find the simple expression for fluctuation. We can be represented using this one. And as you know, this is dissipation. Dissipation in thermodynamic context is very important. And it represented about the dynamical or complicated dynamic here. For example, and it's related to about dissipative structure. So it's mean as a Nobel laureate in 1977, Prigogine, yeah. In a living system, we know about dissipative structure. So it's become a good answer about how entropy and related to dissipation or something like this. I think this is very smart idea for this one. And we try to use this idea to see the surface system, for example, here. And we try to make as simple as possible, yeah, like this one. And now the system model we can derive like this one using this coordination like this one, yeah. And then for the surface area we can representing like this one, yeah. And volume conservation, as you know, we should make sure that there are some conservation for this one. One of this is volume conservation. We make sure for this one. And then for potential function, we can use that. Now we can check the surface for two-dimensional enzyme model. Of course, you can expand to three-dimensional or something like that. At least with the simple calculation, we can see it's work, yeah, for the enzyme model, something like this one. And we can see some surface dynamic here, like this one. Yeah, it's mean including a Brownian motion. We can check, yeah, its work as something interaction between solute and solvent, but in the implicit model. For some people who work in molecular dynamics, there are some big difference, yeah, between the idea for explicit interaction and for implicit interaction. But for some people who want to know just the big something for the trend, not for the precision, yeah, it will be a good calculation. For example, here, yeah, there are some deformation like this one, and we can follow, yeah, what the dynamic. For example, this is for the temperature 37. It's mean typically our body, yeah, typically our body. So we can see the deformation like this one. And we can see for 50 degree Celsius, this is a good, a big deformation for this one. And of course, for 60 degrees Celsius, yeah, this is not a biological temperature, of course, but we can see the temperature effect here can change it means deformation work here. What it mean? Even for the simple calculation, we can show interesting result here. Yeah, interesting result, and how the deformation to the yeah, some geometrical problem. People can follow geometrical deformation, and it's good for preliminary studies for the people who know and then we can check further information by using detailed simulation using all atom or something like that. But at least for people who want to have interest about general problem, they can calculate. This is a very low cost calculation, yeah. So people can still follow just general behavior, yeah. So we can see the effect of temperature on surface deformation for 2D the enzyme model. This is optimum, yeah, and this is denaturation. In the same case, for more than 50 degrees Celsius is denaturation. So it's not good for our biological function or something like that. I think that's all. This is a very simple calculation, but I think for some people who want to know, yeah, the general problem, it's really useful method. Okay, thank you very much.
Well, thank you very much, Mr. Acep Furkon, PhD, for very nice presentation related to the effect of temperature on surface deformation enzyme using Langevin equation. And we know that from this presentation, at higher temperature, the enzyme will experience significant surface deformation. Thank you very much for very brief information for this presentation, Mr. Acep Furkon, PhD. And now we go to the third speaker, Ms. Dr. Yosi Andriani. Good afternoon, Bu Yosi.
Oh, okay, already good afternoon. Okay.
Yes,
Okay.
I don't know, it's already
Thank you. Okay.
And before Dr. Yosi Andriani delivering her presentation, I would like to inform to all of you about Dr. Yosi Andriani.
Okay.
She is a lecturer and researcher in Institute of Marine Biotechnology, University Malaysia Terengganu, in applied biochemistry with educational background. She did his bachelor degree in Sriwijaya University, and then continued his degree for master in IPB University, and graduated for doctoral degree by research in applied biochemistry field in University Malaysia Terengganu, Malaysia. And with her is already more than 20 publication at Scopus for last 3 years. And now to Dr. Yosi Andriani, time is yours.
Okay, thank you, Mr. Moderator, for the timing. Okay, I will share my presentation. Okay, where is it? I cannot find how to make this. Okay, okay, share screen. Okay, can you see my slide?
Yep.
Okay. Okay, how to oops. Okay. Sorry. Oh, wait a minute. How? Okay. Sorry. How to? Okay. Oops. Oh my. Sorry. I cannot find where is the.
We already showed the.
Oh, I'm already so. Okay. Okay. Oh no, not this one. Okay. Okay. Sorry for the technical problem. So my presentation. Okay. Very good afternoon, everyone. Thank you for all. I want to thank you to Mr. Moderator for the guiding the presentation. So my presentation will be in the review on the anti-inflammatory and anti-cancer agents from coastal plants as potential agents for preventing COVID-19 and their future perspective. Okay. So source of this presentation was adopted from the Google Scholar, PubMed, Scopus, and Web of Science in order to find the most recent published articles related to the keywords such as coronavirus, cancer, inflammatory, antibacterial, and COVID-19 pathways, SARS-CoV, herbal medicine, tropical, and coastal plant until 1st of November 2021. Okay. Briefly, a new coronavirus causing acute respiratory syndrome emerged from Wuhan in China in December 2019. Then since then, gene sequencing of the sample taken from the lower respiratory tract of infected patient has made it possible to characterize this new virus called severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2 with familiar name as COVID-19, which given by WHO in February 2020. So the rapid global spread of coronavirus disease, COVID-19, which is caused by the novel beta coronavirus SARS-CoV-2, poses significant threat to public health. So the number of confirmed cases and deaths continue to grow worldwide. Yeah, COVID-19 can develop rapidly into acute respiratory distress syndrome, ARDS, resulting in multiple organ dysfunction of death in some cases. So this is globally, the number of COVID-19 patient has increased by the day before vaccine. Yeah. In April 2020, the figure was almost 3 million case. Then as of March 2021, okay, as indicated by World Statistics, the number increased to 116 million. So with that of total of almost 3 million. So by June 2021, the number of COVID-19 patients was around 173 million, indicating a sub rise. The original death was almost 4 million. So the total voltometers by June 2021. So globally, in order to stop the virus from spreading, most countries have imposed the lockdown, including Indonesia, right? PPKP or PKP, okay, which consequently have a severe impact to the economy. Yeah, yes, maybe lockdown can reduce, but it's consequently have, and it give impact to the economy. Okay, currently a mass vaccination effort have been carried out throughout the world, yeah, including Indonesia and Malaysia, as one of anticipated ways to prevent the COVID-19 pandemic from spreading out. Okay. Globally, the number of COVID-19 patients has reduced by the day after vaccine. So you can see the data from this link. And then this is I give two cases, two sample country, of course, Indonesia and Malaysia. So sorry for the other country. Total coronavirus cases in Indonesia, okay, total by October, 31 of October 2020, we can see the total case is not increased, yeah, but not increased but still stuck here, but not increased at least, yeah, because maybe it is effect by the vaccine. And then for daily, we can see that the cases, daily new cases is significantly reduced. Almost pattern happened in the Malaysia for both total cases and daily new cases. Okay, for the, okay, I cannot find the same data from the Indonesia, so I only put the data from the government in Malaysia. We can see from this graph, COVID-19 death by date of death reduced. Also for the COVID-19 patient ventilated and COVID-19 patient in ICU. So the same pattern for the COVID-19 hospital admission. So reduce, yeah. So doctor is not busy to take care the students like when cases is increased. And then confirmed COVID-19 cases also reduced, and then COVID-19 test conducted also reduced. So we can see that effect the vaccine. However, because the bacteria always transform their genetic to form the new virus. So in another way, besides vaccine, the use of natural ingredient from plant maybe is needed, yeah, in increasingly effort to prevent mortality rate due to the infection by the COVID-19 virus and treatment after infection. So especially in form of prevention due to the inflammation, as well as comorbid disease such as cancer. So di sini for this time, I will focus the inflammation and cancer only because comorbid is correlated to many disease besides cancer, such as hypertension, coronary heart disease, diabetics, and so on. So, but for today, I only focus on the cancer because we have no time for discuss. If we discuss for all comorbids, okay, cancer and inflammation. Prevention of cancer development by non-steroidal anti-inflammatory drugs have been reported by Wong. Some example of this drug include ibuprofen, diclofenac, aspirin, and so on. And then, however, this drug have been shown to produce many side effects to the human. Okay, so therefore, an alternative treatment prevention to reduce the risk of the severe disease caused individual with inflammation and cancer in COVID-19 patient are eliminated. It is critically important to search a new prevention from nature to circumvent the disease to be worse, especially for those already infected by COVID-19. So, medicinal plants have a lot major group 2, and 2 of them have come from the tropical and coastal plant. So, some of coastal plants have been used traditionally to treat inflammation and cancer since many years. However, there is lack scientific evidence regarding this ability correlated to their potential in preventing COVID-19. So, why coastal plant? I said, okay, for personally, I choose the coastal plant because niche area research in UMT is marine. So I choose plant. Usually my project related to the plant. So because the niche area, so I choose the coastal plant. Okay, which still correlated to the plants. Okay, and then why? So another reason is this plant is rich by bioactive compound with many benefits to the human, especially for treating bacterial infection and cardiovascular disease, cardiovascular correlated disease such as cancer, diabetic, inflammation, hypercholesteremia, and atherosclerosis. Okay, then abundant and easy to get. So for the city with the beach, maybe it's easy to get, but for the city with no beach, maybe we can choose the tropical plant, yeah? And not much research compared to the herbal plants, medicine, or tropical plants. That's why I choose the coastal plant. This is list of coastal plant and their potential for preventing COVID. Okay, some of, yeah, there is many, but I choose only a few in this. Terminalia catapa, Lityrus japonicus, Casuarina equifolia, Hibiscus silesius, Pandanus erotissimus, Ipomo pescapre, and Terminalia catapa. And then current benefit we can see based on the anti-inflammatory, anti-cancer, anti-atherosclerosis, and anti-hypercholesterolemia. And I cannot see this. Okay. Okay. From this, we can see the potential mechanism for preventing COVID-19. So why I put the anti-atherosclerosis? Because anti-atherosclerosis is actually is chronic inflammation. So all the same cancer also is inflammation disease. Yeah. So that's and also for the antimicrobial, why I put this antimicrobial plant, because bacteria or microbial is one factor that can cause the inflammation also. Okay, so based on the potency of the compound as anti-inflammatory and anti-cancer, and their group and their gang, anti-atherosclerosis, hyperlipidemia, antibacteria. So we hope they also have potency to prevent the COVID-19. This is potential mechanism for preventing COVID-19 based on the mechanism that I adopted from some literatures. Okay, this is a bioactive compound from some selected coastal plant, as I mentioned before. This is table 2 as potential candidate for COVID-19 prevention, such as Terminalia catapa, Litirus japonicus, Casuarina equivoctalia. Like we can see in almost of the compound from this plant is phenolic compound, flavonoid, and some alkaloids. So that's contribution or study. It is widely reported about the phenolic compound, flavonoid, alkaloid as anti-cancer, anti-inflammatory. Yeah. So this kind of compound probably have a role in their activity. This is some of the structure of compound. I only put some of them. Okay, and next, this is Epomo pascaprae from a plant of Epomo pascaprae and Pandanus tectorius. And this is Terminalia catapa and Soneratia alba or Soneratia cassiolaris. And this is Casuarina ecosifolia and Hibiscus thiliaceus. This is some peak of plants that have potency based on my literatures. Okay, now we talk about the possible pathway. This is first I would like about the COVID-19 and inflammation. Okay, this is potential therapy for COVID-19 by targeting inflammation and cytokine storm with inhibiting NLRP3, BRD4, IVIG, and EL6 as alternative pathways as potential inhibitors such as costarpan. So from this figure is actually under review in my chaptering book, collaborate with 4 universities in Indonesia, including Unsri, Sriwijaya University, and one of the author, Associate Professor Dr. Herman Shah. Okay, from this, you can see that SARS-CoV-2, okay, will it take to the cell by binding the TMRPSS2 and S2 receptor. So I didn't show the abbreviation because it's too much. So my slide is real full. Okay, if someone to know about the abbreviation can contact me. Okay, and then can cause the binding of the SARS-CoV-2 can cause the release virus to the cell and then cause the tissue damage. Tissue damage can produce the neutrophils and produce the NLRP3 inhibitor, BRD4, and the IVIG that can trigger the hyperinflammation and produce the and can release the cytokine storm, cytokine factor that can overproduction. After that, it can cause the overproduction that can cause the cytokine storm. Then that at the end can cause the RRDS, to the COVID-19 patient. Yeah. So by using the inhibitor for the IVIG and RP3 and BRD4, we hope can prevent the inflammation to the, we can prevent or reduce the inflammation to the COVID-19 patient. Okay. The second pathway, the second possible pathway to prevent the COVID-19 infection in patients with cancer disease by using S2 inhibitor and anti-inflammatory potential agent from costalpan and their bioactive compounds. This is also can use for the costalpan usually, tropical plant, I mean. Okay, this is SARS-CoV-2 intake to the cell and been bind to the mediated by S2 receptor, and then okay, can modulate this process to the this process, and then in the last one, then and the last way they will trigger the cancer progression. For this, from this mechanism, SARS-CoV-2 can cause the release of cytokine and then reduce the inflammation. Okay, inflammation can produce the SSP27 phosphorylation that can cause the cancer progression. So from this pathway, maybe we can use a S2 inhibitor agent in this area, and then from this pathway, and then also anti-inflammatory agents. Anti-inflammatory agent can reduce the prevent the SSP27 phosphorylation that can reduce the cancer progression or prevent cancer progression. And then S2 inhibitor agent can inhibit the process that can trigger the cancer progression. This is a possible pathway to prevent the COVID-19 infection in patient. Also, there is another mechanism also, maybe by using the antivirus to reduce virus attached to the S2 also can be possible. Okay. Conclusion: coastal plants can contain bioactive compounds that have the potency to as anti-inflammatory and anti-cancer related to COVID-19 prevention, can be used as a new alternative agents. Okay, future perspective: coastal plant will become more valuable and usable if they can be used as a novel agent to prevent COVID-19. However, further study will be needed through in vitro and in vivo studies. Molecular level investigation on their mechanism can be carried out to ensure that the expectation can be realized. So lastly, when investigating coastal plant, an important things that require more attention in their safeness, especially for people of all age or with comorbidities. Okay, this is some reference. Okay, thank you so much for watching me. And then that's all. Thank you.
Thank you very much, Dr. Yosi, for sharing to us related to the potency of coastal plants for preventing COVID-19. Once again, thank you very much. Now, allow me to invite the fourth speaker, Associate Professor Dr. Zailan bin Siri from Malaya University. Before delivering his presentation, I would like to inform you about the Associate Professor Zailan bin Siri is from Institute of Mathematical Sciences, University Malaya. He did his bachelor degree in computational and industrial mathematics, University Malaya, and master degree in 2004 in numerical analysis, University Putra, Malaysia, and his PhD degree in 2012 in mathematics in National University of Malaysia. Already his research interest is implementation of multigrid algorithm in parallel, natural convection in enclosure, boundary layer problems, and numerical methods analysis. And unfortunately, due to the health conditions, Mr. Zailan bin Siri couldn't present his presentation. For this, his presentation will be presented by Dr. Sarifah Nuriza. Dr. Sarifah, time is yours.
Hello, good afternoon. So in this presentation, I will be talking about the work that Dr. Zailan and I do. Just give me a moment to share my screen. of everyone. Right. Is everyone able to see my screen?
Yeah, we can see it.
Okay, great. Right, so in this presentation, I will be talking about Marangoni convection in a C-shaped enclosure with partially heated walls. So before I jump into the actual problem itself, I'd like to go through the basics first and talk a bit about Marangoni convection. Now, Marangoni convection is a natural convection, and it's a type of phenomenon where heat and mass move to an area with a higher surface tension due to changes in the surface tension gradient. Now, the surface tension gradient is greatly influenced by temperature. So if temperature is high, then the surface tension gradient would be low. And similarly, if the temperature is low, then the surface tension gradient would be high. Now, why is this important? Well, it's got a bunch of different applications, and one of them is crystal growth, and the other one is electron beam melting. Now, electron beam melting is this process where you take metal powder, and what you can do with it is that you can melt it and you can make mechanical components out of it, and that involves Marangoni convection. The second bit that I would like to talk about is the C-shaped enclosure itself. You might be thinking why this was chosen, and that's because, again, it's got a number of different applications. And the first is electronic cooling. You could place a bunch of different electronic components in a number of different places so that they do not crowd one another. And the second is heat exchangers, where it's a system where heat is exchanged between two or more fluids. And this is a real life example of that application here. You've got the heat exchanger on the right side here, and it is submerged in a water storage tank. And what it does is that it exchanges heat. Simple as that. Moving on to the problem itself, we have here the cavity itself. On the vertical wall here on the left side, we have it's heated by temperature TH, and similarly for the bottom wall, it's also heated by temperature TH. The top wall here is where Marangoni convection takes place, and hence this equation at the top, the shear stress equation. This C section here is where the cooling bit is. So that those walls are cooled by temperature TC. And these two walls here, FG and DC, they are adiabatic. And that just means that the temperature gradient themselves are zero. So heat is not gained or lost. Here is, these equations here are the governing equations that describe fluid flow and temperature within the cavity. So these are dimensionless, by the way. In the first line, you have the continuity equation. In the second and third line, you have the momentum equations. In the fourth line, you have the thermal energy equation. And in the fifth line, you have the stream function. Now, the stream function is used to plot the streamlines, which will give you the trajectory of the fluid elements themselves. Some people might say that this looks nasty. I think it looks nice, but again, it depends on the person, of course. Here are the boundary conditions. I've addressed them before, so I'm not going to go through them again, but here they are. Now, before I get to the results themselves, I'd like to talk a bit about the parameters. There are quite a few here. And the first one is the Marangoni number, which goes from 0 to 1,000. The second is the Rayleigh number, which pretty much gives you an idea of how laminar or turbulent the fluid flow is. So that goes from 1,000 to 10,000. And then you have the depth, which is the depth of the cavity, that goes from 0.3 to 0.7. And then you have phi, which is the solid particle volume fraction. That is the percentage of nanoparticles that is mixed with the fluid. So that goes from 0 to 0.3, or 0 to 3%. 0 indicates the absence of nanoparticles. So the results themselves. Here we have the streamlines for RA close to 1,000. I'm not going to go each and every single one of these because that would take too much time, but the general idea behind this is that as the Marangoni number increases, you can see that the single cell, when natural convection is absent, splits into two and moves to the top where Marangoni convection takes place. And the fluid flow themselves become stronger as NA gets bigger. Similarly, if the depth of the enclosure is increased as it gets bigger, you can see that the cell themselves get bigger and fluid flow, the strength of the fluid flow also increases. Looking at the isotherms, when Marangoni convection is absent, when it is 0, there's barely any sort of deformation at all within the temperature contours. However, if you increase MA, you can see these slight distortions here. And as the cavity gets bigger, the deformations themselves get even more prominent, and it looks as though the temperature itself is moving up based on this picture here. Moving on to the next bit, which is the average Nusselt number. Now, the average Nusselt number is the convective to conductive heat transfer rate array, and that pretty much tells you that if the value is bigger than 1, then convection is dominant. Less than 1, then conduction is dominant. Tx here represents the vertical heated wall, Dy represents the horizontal heated wall, Tx plus Dy is the total average Nusselt for both walls. Now, looking at the vertical wall, we can see that it looks like the average Nusselt increases linearly with the Marangoni number, which is exactly what we need. However, regardless of the value. However, if we now look at the horizontal wall, you can see that it looks at least when the cavity is the depth of the cavity is 0.7, it looks as though the numbers themselves are constant. It's not moving at all. And if the depth itself gets bigger, it seems as though the heat transfer rate is decreasing. It's not doing too well. If you then combine both of these values, it looks like the average Nusselt increases linearly again, as with the vertical wall. So that suggests that the horizontal wall is doing also well, is doing all the hard work. It's coming from the vertical wall.
Sorry, Dr. Saliba.
What can we do?
Saliba.
What can we do? Well, the first is that the strength of the flow increases not only with the depth of the cavity, but also with the Marangoni number and the Rayleigh number. The second bit is that the highest values of the average Nusselt are registered by the enclosure with the smallest depth, which is 0.3, and this is likely due to the hot. The final bit is that the Marangoni number has really no effect on the horizontal heated wall, at least for when it's less than 0.7. Or bigger than, for when it's bigger than 0, when it's equal to 0.7, it decreases the average Nusselt. And that brings me to the end of the presentation. Thank you.
Thank you very much, Dr. Sharifah, for quite information regarding Marangoni number. But unfortunately, I couldn't hear your voice clearly. Once again, we will discuss it further during question and answer session. Now, last but not least, allow me to invite the fifth speaker, Ms. Endang Linirin Widyastuti, PhD. Good afternoon, Bu Endang.
Good afternoon. Thank you. But let me share my
Okay.
screen first. Okay, thank you.
Before,
Thank you. Yes.
Linda,
Yes, sir. Yes,
before
sir.
you deliver your presentation, I would like to read your
Oh,
curriculum.
yeah.
briefly.
Sorry.
Okay.
I'm sorry. I so eager. Anyway, I stop screen. Thank you.
Uh, Ms. Endang Lenirin Hudastuti did his bachelor degree in Indonesia University of Education, Bandung, and did her master and PhD degree in University of Kentucky, United States of America, in biological department. And Ms. Endang, Dr. Endang, from Department of Biology, Faculty of Mathematics and Natural Sciences, University of Lampung, and also she is head of Coastal and Marine Research Center of Research and Community Development of University of Lampung. And now, Ms. Endang, time is yours.
Thank you. Thank you so much. Let me share my screen. Okay, thank you. But before I delivering my research, let me have appreciation first to the rector of the Sriwijaya University, and also to the dean of the Math and Natural Sciences, and also my sincerely thanks to the organizing committee of the SICBAS. Anyway, I have to correct the last one. Previously, I'm a head of the marine and whatever you call it at the last one, but I'm not longer working on that on the session. But anyway, okay, let me introduce my talk. Okay. This one is about taurine and its physiological function. I did this, it's about 20 years ago. Since 20 years ago, I've been working with the taurine. And this is the content of my talk. I will have a briefly introduction about taurine. I think everyone knows that. But anyway, in order to start with the talk, let me introduce with the taurine first. And then the second time, I would like to express all the experiment that has been done by using the taurine, and finally with the summary. So the taurine itself is a free amino acid, and everyone knows that. And at the beginning is sort of like from my dissertation, I found that the renal development of the Rhinacatis biana is swollen because of the osmotic stress. And finally, we tried to look at the content of the swollen cell, and then it seemed like there is a lot of organic osmolyte. One of them is taurine. Therefore, after I'm coming back to Indonesia, I tried to apply the taurine, sort of like to improve the survival rate. At the first time is with the Peneus larvae, but unfortunately we cannot publish the data because it is a part of the data is belong to the company. And next, I tried to also to give to the grouper fry, and also improve the survival rate of the grouper fry. So these three things that they come up with my idea. Maybe taurine is really important for any other physiological function in a human. So I move on with the anti-diabetic, something like that. But before that, there's some research also is conducting with the anti-obesity by reducing the plasma liver cholesterol, and also proving that the taurine also prevent the NAFLD is a non-alcoholic fat liver disease. And also other study show that there is a taurine also able to reduce the fat deposition. So the antibiotic process that I was thinking about, that maybe that taurine can have any particular effect on anti-diabetic. So we use the sort of like putting the animal into status of the diabetic by injecting the alloxan. And then it seems when we try to measure the glucose level, and also we look at the kidney morphology, it seems like the taurine be able to decrease the glucose level and also able to repair the kidney damage. And finally, we try to look at the taurine function as antioxidant in cellular level. And before we move on with all the experiment that we have already done, other study has already mentioned that the taurine also modulate the red blood cell as an antioxidant metabolism, and also taurine indicate the anti-apoptotic and anti-therogenic properties. So this is the source of biosynthesis, but I'm not going to go through with it, and just only to see that this is the mechanism of biosynthesis of the taurine. Basically, everything that from metabolism of levocystine by using oxidation can turn into the hypotaurine, then hypotaurine can turn into the taurine. Taurine itself is a major product of the sustained metabolism in the mammalia, and recently marine bacteria also proved that they can contain of taurine biosynthesis. They can have a biosynthesis taurine biosynthesis pathway. So anyway, if you look at this SICBAS on the Tafetia et al., she mentioned that there is at least three kind of a mechanism in order to produce the taurine through the biosynthesis. For example, with the microalgae, can goes to the taurine production through the serine, and then converting the serine into the taurine. And then in animal tissue, by using the carbon CDO, they can turn the cysteine into the taurine itself. And also the last one I have already mentioned about the bacteria. But anyway, so this is a good thing of us since we knew that the coastal line is, so the previous speaker have already mentioned 70% of the earth is an ocean. Maybe a lot of is a potential for us to do something with the any biota in the ocean, particularly with the microalgae. The video also proved that the increase in the salinity with for the microalgae can elevate the taurine level. So this is maybe giving us an opportunity to work with it besides using any microalgae that can be used for the biofuel. And I come up with, I'm searching in the Google, and it seems like the taurine content on the food is very high, particularly with the cheese, and then in the cottage, it has like for like a 3 ounce or 1 cup contain of more than 1,000 milligrams. So anyway, we can find this taurine in a daily, our daily meal. So now the second part is how I can run, how my research run with the taurine. First, we use it in the fresh water, and then also later we use the seawater. But I'm sorry, first. The first one is use the seawater because at the beginning we have already know that taurine can be used as osmolalate organic. So maybe we can have something to learn that taurine can have any physiological function, or maybe they may affect the metabolism transmitter. For example, this is already done by other research, and surprisingly, after we put the taurine into the plankton, we sort of like enrichment the plankton and then giving to the grouper fries or Epinephrolus fucogatatus, the survival rate of the grouper is increased. It's more than 80%. So we try to also to look at the other functioning of the taurine. And the next is we use the taurine in the cobia. This is very surprisingly, we use the juvenile taurine, juvenile cobia or Rhacicentron canadum, or in Indonesia now we is famous with the king cobia. We use the Lysocentron canadum juvenile when we put them with a different treatment and different meat. Like for example, one group is with the squid, only squid, and then the other group with squid with the taurine, and then the other group with the commercial fish food, and then commercial fish food with taurine. It seems the gonad weight is increased those group that given the taurine. So if the gonad increase in the weight, and we think about whether there's any maturation of the gonad or not, or maybe just because the weight of the gonad also correlate with the increase in the body weight. But yeah, we try to measure the estradiol, and it looked like every meat for the fish that we put the taurine increase in estradiol. So we could say that probably maybe it's too early, but maybe this need further investigation. So maybe the taurine also can improve the maturation of the gonad in the seawater fish. So later we use the freshwater fish, and then we use the cobia and gurame. And this one is have been done in 2011, and it seems every group that we give the taurine inside for their food increase the body weight, the growth weight for 25 to 60% compared to the control. So those two things that we hypothesized earlier, that taurine maybe affect the physiology of the fish, either in the freshwater or in the seawater. So next thing is we want to see whether there is an anti-diabetic effect of the taurine itself. We inject the mouse, we use the male mouse, and then we inject that with the alloxan in order to give the effect of diabetic. And then after that, we give the diet. And from here on the right side, we can see that the reduction of the glucose level is much higher to those that in the group that giving with the taurine, as well as the kidney, because we also measure the destruction of the kidney, and the score of the kidney repair also is very good. It's very significant compared to the group that only given for the alloxan. So here in this also we can say that the taurine, giving a taurine for 40 days after injection with alloxan can reduce the blood glucose level, and also can repair the kidney damage caused by the diabetic effect by the alloxan. Finally, we try to look at the antioxidant effect, but this one is by inducing the male mice. Also, we use the male mice as a sample animal by using the benzoylpyrin, and we look at the blood profile, either the erythrocyte and also the leukocyte. From here, we can see also the data. Those that given with the taurine also has an effect of ability to getting back to the normal for either for the erythrocyte and also for the leukocyte. And we also try to look at the different count, differentiation count for the leukocyte. It seems like the neutrophil is also affecting the number of the neutrophil, also affecting by giving the taurine. Finally, we try to test any effect of the taurine for the anti-cancer. So we use the HeLa cell line, and then we try to compare that with the doxorubicin, that means mostly is very common for the cancer drug. And we look at the cell viability, and this one is using the MPT assay. And it seems like the taurine also has the effect on the cell viability. We try to look at with the different, this one with the higher concentration, this one with the lower concentration of the taurine. It seems like the doubling time of the HeLa cell line is also affecting by the giving the taurine. The control, for example, the control have a doubling time for 72 hours, but by putting the taurine, giving the taurine in the cell line, and they increase the doubling time. So it means there is some resting of the proliferation of the HeLa cell line. So we try to look at this, and then maybe assuming some of the HeLa cell line is inhibit to proliferate or causing the apoptosis by the taurine. We try to look at the IC15 for giving the taurine. It's high, so it's not toxic actually, but it can cause the inhibition of the HeLa cell line. This is the morphology of the HeLa cell line with a different treatment. We come out, this is a control, and then this is using the doxorubicin, and this one is using a taurine. It's still, some of them is still there, but some of them is already also undergo the apoptosis. Presumably is apoptosis. So we try to look at that in the P50 expression of the P53 gene. And unfortunately, we just have the data. Presently, we haven't calculated yet. But from the RT-PCR by using the reference gene GADPH and also primer 53, there is a expression of P53. But when we use this one, the oldest one, we use the concentration of the taurine much less than the previous one. So it's about, we use a 50, 75, 100, and 125 ppm. It looked like there is also expression of the P53. So therefore, we can say that taurine can cause the apoptosis of the HeLa cell line through the expression of the P53. Actually, the next thing that we have to run from experiment is maybe we have to measure or to look at the expression of the P21. So if that we can collect all the data for that, then we can come up with the idea that taurine can be used as an anti-cancer. So the summary of my talk, first of all, we knew that the taurine, the amino acid, the free amino acid, is one of the organic osmolyte, as can be accumulated in the animal when they suffer from the osmotic stress. Second, taurine also can improve the gonad maturation and also the growth rate of the seawater or a freshwater fish. And third, taurine can also have an anti-diabetic effect. And the fourth, taurine also have an antioxidant, and particularly for the anti-cancer by inducing the apoptosis in the cancer cell line. I think that's all for my talk. I'm sorry, I tried to make it simple. But anyway, I would like to express my knowledge because all the research is being done because the funding from the Ministry of Education, Culture, Research and Technology. And also we thank to the Sea Farming Agency from the Minister of Marine Affairs Fishery. And we also thank to the Faculty of Medicine Pajajaran University for helping us to run with the real-time PCR and also for the MTT essay. I think that's all. Thank you for time and for your attention.
Well, we do thank you to Ms. Endang for such great presentation and also very complete information related to taurine as amino acid and its physiological function, including I see on the slide taurine biosynthesis in microalgae, fresh and seawater fish. We do thank you very much for once again. And those are the presentation from all the speakers. And now we're going to the next session, which is a discussion question and answer. If someone may have a question, just raise your hand and ask to the speakers directly. Is any participants may have questions directly, can give directly to the speakers? No? Okay. All right. I will read the question from all the participants already typed in on chat room. First, from Desnally to Dr. Dede Heri Yulianto. First question is, what is the prospect of using WRF for textile wastewater degradation if applied to industry, based on the synthesis process, time, cost, and effectiveness of degradation? And the second question to Dr. Dede Heri Yulianto from Rian Astri. Is there any possibilities of developing biomaterials not only as absorbents but also as photocatalyst materials? Also, where will biomaterials research lead in the future? To Dr. Dede, time is yours.
Okay, thank you very much for the question. I will answer the question from Desnally. Thank you for the question. So vital fungi could effectively degrade the most of synthetic dyes and also most of textile waste water, based on my experience, of course. And the production of this water fungi or mycelia is also cheap. According to our calculation, we can produce only around 600,000 rupiah for 1 ton of the mycoleca. So it is a very cheap compared than other chemical synthetics or other preparation for absorbent materials. And also, according to the times, yes, this water fungi has the ability to degrade the synthetic dyes in the wastewater vary from 3 until 3 until maybe 30 days. So therefore, in this technology, we try to combine between water fungi and also other materials. Not only super absorbents, for example, but also as a when Rian Asri asked about the possibility using the photocatalytic material, it is also maybe can be used. So yeah, this is possible to use this combination with the water fungi. And also about the, what else? Yeah, maybe this is the question for both question.
Thank you very much, Dr. Dede, for the answer. Second one, questions for Dr. Yosi. Question to Dr. Yosi from Fernanda Halheti. It was mentioned that several compounds with certain activities have the potential to be anti-COVID. What is the relationship between compounds that have anti-tumor activity as well as potential as anti-COVID? And the second question to Dr. Yosi from Indah Soliha. Are there any studies comparing the phytochemical components of plants that live on the coast with other areas with different altitudes? Time is yours.
Okay, thank you, Mr. Moderator. Okay, I have two questions. The question from Felina Hayati in Nature. I'm not sure, Dr. Felina. Okay, it was mentioned that several compounds with certain activity have the potential to be anti-COVID. What is the relationship between compounds that have anti-tumor activity as well as potential anti-COVID? Okay. Okay, jadi she asked me correlation between compound with the anti-cancer potential activity with the and its correlation with the anti-COVID potential activity. Okay, based on the SAHA, okay, I would like explain according to SAHA and Anirfan, they have suggested two possible ways of the cancer progression correlated to inflammation in the COVID patient. What the correlation? So first, they use anti-inflammatory and anti-cancer. Yeah. So according and then S2 okay. It is widely known that S2 mediated virus entry to the cell, and then S2 ACE2 upregulation is associated with the reduced cell proliferation. So which indicate that S2 is a potential protective factor for cancer progression. Yeah, virus entry to the cell mediated by S2 receptor lead to the intake of S2. The more S2 receptor available in the membrane, the more virus can enter the cell. S2 inhibitor can prevent virus entry to the cell. However, a decrease in S2 receptor in the membrane may result, it can may result in increase the level of angiotensin 2 type 2 or AT2, which trigger inflammation of cytokine release, thereby causing acute respiratory syndrome, heat shock protein SSP27 phosphorylation, then can induce cancer progression. So why I use correlated to the cancer? Because some compound that have anti-cancer potency based on their pathway, actually there are some pathway correlated to the anti-cancer compound. Then the same pathway is correlated to the if H2 is reduced in our cell. So correlated to the imbalance in RAS, bradykinin elevation, ERK1 and 2 map, and so on. Then because the H2 is reduced, so because the much cell intake to the H2 are mediated, so it causes the factor that can induce the cancer progression. So by use the anti-cancer compound, we hope that the process that can cause the cancer progression factor will reduce. So thus we use anti-cancer because some of the mechanism correlated to the mechanism that affect by the SARS-CoV-19 intake to the cell. Okay, do you get it? Okay, inshallah. Okay, so this is our possibility. So however, we need a further study to prove this possibilities. Okay, the second study is, are there any studies comparing the phytochemicals component of plants that live on the coast with other areas with different altitudes? Okay, I'm not sure there is a study or not, but I think not much study correlated to the coastal plant, but for the tropical, maybe yes, many there is any, but you can do comparison study on the phytochemical component of plant that live on the coast, maybe coastal plant from the Terengganu area compare with coastal plant with the Bengkulu area, maybe with the beach area. You can do it because some of study reported that there are geography and sampling area, different sampling area can be effect to the compound that plant produce. Okay, and also activity. Okay.
Thank you very much, Dr. Yesi.
From Indah Soliha. Thank you for the question.
Okay, thank you very much for Dr. Yossi for answering the question from Ferlina Hayati and Indah Soliha. And the second chance from Siska Otarina asked to Dr. Sarifa, even though I saw that already answered in chat room, but the question is, you have used finite element method to solve the governing equations. Is it possible for us to solve the equation by using another optimization methods? Dr. Sarifa.
Yes, Yeah, okay.
Thank you very much.
So it's very much possible for you to use any other method to solve the governing equations. That's completely fine. The other two options are finite difference and finite volume. But there is a catch with finite difference in that it only works well with structured domains. And what I mean by that is that it only works well with squares and rectangles. It doesn't work all that well with, say, a circle. That's its limitation, unfortunately. But yeah, it should pretty much work well with finite element and finite difference or finite volume for a problem like mine, or as long as it's structured.
Okay, thank you very much, Dr. Sarifa, for double explanations.
All right, that's fine.
Thank you.
Thank you.
And I think the last question is from Saum Siam, Pharmacy, Sriwijaya University, to Dr. Endang. The question is taurine also potential in inhibiting oxidative stress in type 2 diabetes? Focus on the mechanism of oxidative stress. Thank you.
Okay, thank you. I guess so, because taurine is from the data we already seen that the taurine also has an antioxidant, so it can absorb all the free radical that cause the oxidative stress of it. One of I'm sorry, I didn't present my research because one of my students also conducting some research, and it's about using a taurine for the for oxidative stress. I forgot or where that he is published about this research, but yes, for sure, the taurine can be used for the to inhibit the oxidative stress in the type 2 diabetic. I guess that's it. Maybe I have to give some information. Two of my students also joined for this conference, and maybe after this live talk, they're going to give the presentation. Thank you.
Well, thank you very much, Dr. Endang, for the answer. And ladies and gentlemen, we are finally come to the end part of this session from the invited speaker session. Once again, we would like to thank you to distinguish all of the invited speakers who already shared to us beneficial and meaningful act for our future lives. Once again, thank you very much. And before closing this session, welcome to big applause to all of our invited speakers today.
Thank you.
Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon.
Waalaikumsalam warahmatullahi wabarakatuh.
This is the end of the plenary session. The next agenda is break time. To all audiences, please take a break and enter each room at 13:15. Please keep in mind that meeting ID and password for the main room and the parallel room are different.
Kampung halaman selalu mengirimkan anginnya agar membawaku kembali. Di sini, di Sumatera Selatan. Provinsi yang telah maju pesat berlari bersama deru laju zaman tanpa mencabut akarnya. Provinsi yang memiliki gelombang air yang bersahabat, tanah yang kokoh, dan angin yang menari-nari. Sekarang izinkan Kulo membawa Niku menelusuri Sumatera Selatan. Diberkahi dengan kondisi alam yang beragam, Sumatera Selatan merupakan arena bagi siapapun untuk menjelajah. Tempat mencari sumber hidup, lahan bermain, dan berbagai petualangan yang menanti untuk ditaklukkan. Banyak yang bisa dilakukan di sini. Sejak dahulu, Sumatera Selatan merupakan rumah dari peradaban-peradaban yang silih berganti, menempati dan meninggalkan jejak kehidupannya. Meninggalkan kebanggaan bagi generasi selanjutnya, bahwa leluhur mereka merupakan pelaku sejarah yang hebat. Merupakan medan perjuangan bagi para pahlawan dalam menopang keutuhan negara. Beberapa dari jejak sejarah tersebut kemudian menjelma menjadi sajian pertunjukan dan seni budaya yang mengunggah rasa dari tiap-tiap yang mendengar, melihat, dan ikut larut dalam alurnya. Di sini, siklus kehidupan berputar stabil menopang kebutuhan masyarakat. Di sini juga, adat budaya dan perkembangan zaman yang tak bisa dibendung melebur saling menguatkan. Aktivitas perniagaan di setiap sendi tidak pernah berhenti bergerak. Ia menjawab apa yang dibutuhkan oleh setiap insan untuk memutar roda kehidupan. Aku suka ungkapan puitis seorang sufi, Maulana Jalaluddin Rumi. Ada cinta di setiap agama, namun cinta itu sendiri tidak memiliki agama. Tiap-tiap keyakinan saling berpegangan tangan, menguatkan dan menjadi pilar yang membantu Sumatera Selatan berdiri tegak sejak dahulu kala. Menjadi pedoman hidup dari satu peradaban ke peradaban lainnya. Sumatera Selatan juga merupakan rumah berbagai event-event berskala nasional dan internasional. Dari event kebudayaan dan tahunan, turnamen-turnamen olahraga tingkat dunia lainnya. Semua kegiatan penting itu dapat berhasil berkat dukungan dari masyarakat dan ditunjang oleh fasilitas umum dan infrastruktur yang memadai. Pemerintah Sumatera Selatan senantiasa menyiapkan fasilitas yang dapat dimanfaatkan oleh masyarakat agar tiap individunya dapat bergerak maju tanpa hambatan. Ayo bergerak, ayunkan kaki dan tangan, tantang angin, lawan arus air, bukan panah yang bebal. Raga yang prima, jiwa yang kuat, dan mental yang sehat mencerminkan kualitas individunya. Ayo bergerak, tuangkan ide, dan buat sesuatu. Asa cipta hasilkan karya. Provinsi ini memiliki tangan-tangan bertalenta di setiap generasi dari berbagai sudut daerah. Mewujudkan dan melestarikan gagasan karya para leluhur, sambil tetap beriringan dengan laju zaman. Dan sungguh leluhurku adalah para peracik yang hebat. Kekayaan alam Sumatera Selatan dapat diolah menjadi berbagai kudapan yang menggugah selera. Inilah provinsiku, kampung halamanku, rumah bagi semua orang, rumah bagi berbagai penutur bahasa, etnis, pemeluk agama, dan berbagai individu yang memiliki keyakinan untuk maju.
Waktu dan laju zaman adalah hal mutlak yang akan terus berjalan. Mendesak setiap insan untuk bergerak maju ke depan.
Provinsi Sumatera Selatan sendiri telah diberkahi sumber daya yang melimpah dan beragam sebagai modal untuk dikelola demi terciptanya kemajuan di semua lini dan untuk semua individu yang hidup di dalamnya.
Pemerintah Provinsi Sumatera Selatan mengajak setiap insan Sumatera Selatan untuk bersinergi, berinovasi, bergandengan, mewujudkan kemajuan di berbagai sektor. Karena kemajuan dicapai oleh kita dan untuk kita sendiri. Sumsel maju untuk semua.
Zoom Zoom ditutup. Bapak Ibu, Zoom kita ending. Terima kasih